Okay, so just to remind you about CAR T therapy. So in CAR T therapy, you draw the T cells from a patient, you draw the blood actually, then you separate the T cells, and then you engineer the T cells. So you add the DNA, and we talked about how you can add the DNA to the cells. There are many, many different ways. And then the DNA makes the protein, and this protein is actually CAR, chimeric antigen receptor. And then once it is expressing, you where is number 3 here? 1 2. Then you make tons and tons of CAR T cells, you expand them, you infuse it back to the patient, and then these are ready to recognize the antigen and kill the tumor cells. Okay, so for CAR T things, which T cells do you use? So usually you use a 1:1 ratio of CD4 CAR T cells and CD8 CAR T cells. Okay, so you use 1:1 ratio. These are also called as helper CAR T cells. And once, see, these are the chimeric receptors. So they latch into these tumor cells, and then they will release these perforin, cytokines, granzyme. Granzymes are serine proteases. So perforins create pores, and then granzymes get inside. It will chew up all the protein. The tumor cells will die. And once they die, they release all the antigens. So other uh, immune cells are recruited in that area, which will clean up and attack the tumor cells furthermore, cleaning up the tumor. Okay, so just to give you a little bit idea about CD4 and CD8 cells. So these are both T cells. So they have found, scientists have found that 1:1 ratio of CD4 versus CD8 is the best combination. So CD4 plus CAR T cells provide cytokine support and has CD8 functions, and they are helper and cytotoxic. They secrete these ILs, and CD8 are also called as cytotoxic tumor cells, T cells. So they are involved in direct tumor killing. They release perforin and granzyme, which leads to rapid tumor death. So usually 1:1 is the best ratio for enhanced efficacy and improved persistence. You want these guys to be there for a long time. So the CAR T therapy is good in a way that these T cells are in human body. Once they're infused, although it is very expensive, we talked about it yesterday, but once it is there for a very long time. So now I'm going to tell you a lot of fun stuff. So this technology, once it is was made, then people are making all sorts of combination. Okay, so these are special variants, and these are not all the variants, just a few variants. There are a lot more than these, but this will give you an idea. So if you take a simple CAR, so simple CAR is this, and this is, as you know, this is an antibody. So this part, so in the antibody, this is called the variable part. This is the part which recognizes the antigen. Okay, so this part and this part. So this part is equivalent to this part in CAR. This is the one which will recognize the antigen, and then all of these things are there, because if it just binds and does nothing, it is useless. So these domains are added so that it recognizes, and then it transmits the signal to kill the tumor cells. That's why you have these domains here. Okay, so first kind is tandem CAR, or short it is called TAN CAR. Okay, so look at this card. So this is so tandem means it recognizes two antigens simultaneously. So if it recognizes two antigens simultaneously, the binding will be tighter and it will be more specific. So look at the CAR here. So it has two parts to recognize. So usually it has only one part here, right? But look at this thing. It has one part and another part. And these two parts are recognizing two different antigens. Okay. So the important thing is that how do you differentiate between TAN CAR and, so this one is not a TAN CAR, because this is a CAR T cell, but it is expressing two different receptors, all together two different receptors. So you can imagine one of this here, one of this here. This is not tandem, this is bisistronic. So I am one CAR-T, but I'm expressing two different receptors, one for this antigen, one for this antigen. That is bisystonic. But if there is only one CAR, but it is recognizing two antigen, that would be tandem. That's the difference. So that's what is shown here. So look here. This is one CAR, but it is attached to two different recognition domain. So this is tandem CAR 1 and 2, part of one only. This is bisystonic. Although CAR-T is one, the cell is one, but it is expressing two independent receptors. Okay. So, so this is stand-up CAR-T. So what happens is that anytime you may even make a CAR-T, right? So tumor will try to hide that neoantigen, okay? Because it knows if the antigen is expressed, it will be killed. So if you have two recognition sites, the second one will still latch on, right? So that's the advantage. So tandem CAR recognizes two antigens simultaneously. See, first part, second part. This is TAN CAR. You can even make three antigen specific. This will recognize one antigen, second antigen, third antigen. So triple. Now this is not TAN CAR because the same cell is expressing two different ones, the whole thing. This is called dual CAR. This is called triple CAR. But if there is one receptor altogether, but different domains which are recognizing, that will make it tandem, yes. Yeah, so here for simplicity's sake, they are showing that there is hardly any linker, but usually they provide different linkers. And when you are developing this thing, what you do, you try many different combination. You change linker length to see. So this is a very important thing whenever you make tandem. So suppose this is the tumor, right? And you are making two antigens specific, but one antigen is here, one antigen is here. And if your linker is not flexible enough here, it is not going to bind here, right? If it is next to it, and if the linker length is flexible enough, then it will bind here. So that is always the case, that how do you know what is the exact linker length? But you have to give it some flexibility so that it can see the whole surrounding area. Having said that, what people do, that some people say, oh, we are making a TAN CAR where it will get activated only when it binds both the antigens. But then the problem is that it binds one antigen, but the second antigen is not in close proximity. So this is rendered ineffective, right? So it is better. You will obviously get more specificity if it is able to recognize two tumor-specific antigens, right? But that is also comes with a risk because they may not be next to each other or your linker may not reach it. A linker is way too flexible to just find something next to it, right? So usually it is better that it works even with one recognition. Make sense? But you have to play with the, anytime you make tandem kind of stuff, you have to play with the length of the linker to identify the best linker. Okay? So two linked, these domains are called SCFV. So two linked SCFV domains, like this is again CD19, CD20, then rest of the things, they remain the same. So advantage is that only one CAR construct, it is relatively cheaper to make and easy to manufacture, less risk of tonic signaling than two separate CARs, strong synergy when both antigens are present. Okay. Now dual CAR, as I said, dual CAR is this one. This is not. Here you are putting two different ones at the same time. That is co-administration. You're giving two drugs at the same time. So this is not dual CAR. You are just administering two independent ones simultaneously. Here also you are mixing them together. See, all of them are separate. But here this is bisystonic or dual CAR. The single one is expressing two independent receptors. Here the advantage is that they are likely to bind. But here you have to also play with the concentration of the dual CAR, right? If you use too much of it, then every CAR will just bind one of the antigen because you have huge excess of it. So you have to play with the concentration of this one so that both of them are used at the same time, right? So keep in mind, one car will actually not have only two copies. They will have multiple copies. So you have a choice of getting both of them latched on if they're at the right distance. Okay. Then this is very good, modular CAR-T. This is very, very good way of doing it. And you will see why. So what happens is that tumors are very uh, adaptable, right? They keep changing their tumor antigen. So you spend 500K in dollars, right? $500,000 just to make a CAR-T therapy against an antigen, and that antigen, the tumor hides that antigen after some time. So the whole money just basically goes as a waste. So this is a really awesome way of addressing that. So modular CAR-T is an advanced form of immunotherapy. So you look at this thing. So this is the tumor cell, and these are the antigens, the black ones. Okay. Forget about this part for a minute. Okay. This is the CAR-T here. Okay. So this CAR-T will recognize this green circle here. Okay. So you can make the green circle separate. This is not directly attacking the tumor cells. It is only binding this thing. And then based on the antigen, you can change this structure. Okay. So imagine, let's give the analogy of the Gyan course, right? You are all the students and I am the teacher who is teaching it, right? But I did not know any of you and I had no access to any of you. And no. So Venkatesh is the middle person who knew me, who got me, and he's the person who recruited all of you, right? And if he organizes another grand course, he will recruit other students and same teacher or a different teacher. So he's the one who is coordinating between you guys and me, right? So this part here is Venkatesh. You guys are whichever one you want to choose. One of us is here and all of you are, you understand the point here, right? One part, second part, and the middle person, right? So the beauty is that Venkatesh can choose whoever he wants to choose as a teacher. He can choose whoever he wants to choose as the students, right? So this is very versatile method. So what happens is that this part is fixed. You make a CAR-T. And you don't have to go for which tumor antigen, nothing. Everything is ready off the shelf. And now tumor cells will try to be very smart. They do different kinds of things. So you just change this. This part is ready because this part will bind here. This part is ready. You just change this part according to the tumor antigen and it will work every single time. Is it making sense? So this is shown here. So in between you have CAR-T and these are all tumor cells. And then, so you give this modular compound. Okay, so this is the CAR-T here. Okay, and then you give the switch molecule. So on one end, it will bind this one. So let's look at the green one here. So green one is binding the CAR-T receptor, and the top part is recognizing the tumor antigen. Okay. They are small molecule, relatively small. Let me see, tumor antigen. No, it will not be a small molecule because it has to recognize the antigen. So it has to have a whole receptor-like a structure to identify the antigen. So it would not be a small molecule because they are not receptors. If they are receptors, then it can be a small molecule. So for example, if it is HER2 receptor, definitely small molecule there. So it can be a small molecule. Most likely it is a small molecule because these tumor antigens are not displayed. So what happens is that these tumor cells are intact cells. So they are not, most likely you are targeting the receptors there. So you can always use the small molecule like folate there, right? So it is definitely a small molecule. So this is the beauty here, right? So you are recognizing, and CAR-T, of course, is a receptor. So small molecule link together. Okay, so if you use low concentration, you kill one cell, then more and more you kill all of this, right? Makes sense. Now this is even better. Okay. So look at here. Look at these tumor cells. I'm showing two of them. Even in this one, they have different receptors, the blue receptors, the black receptors. And you can also think the tumor is heterogeneous. So someone will have blue receptors only, someone will have black, somebody will have yellow receptors. All different kinds of receptors, right? So you add this compound. So this green part is engineered to recognize the CAR-T, and this pink part will recognize this blue receptor. So this one dies. Okay. Now here, this one is blue receptor. This cannot recognize. So this one will not die. But then you switch it to a different one. See, you switch it to different one. So now this one will die. If you use both, now both populations will die. So the beauty is that your CAR-T is the same. Okay, CAR-T is the most expensive part to make. You are just switching molecules. As tumor switches, you switch the molecule. And linking two small molecules is a piece of cake, correct? Okay, yeah. The chances are CAR-T therapy is so good that when you start it across for some antigen or some receptor, it will kill it. But suppose it develops resistance, or if the tumor is heterogeneous and you do the biopsy, you already have 2-3 things in hand, or maybe 5-3 things in hand. So you can easily make this switch molecule, right? Because these are two small molecules linked together. So you can easily make it, okay? Now keep in mind that suppose this CAR receptor does not bind a small molecule, it binds something, but we know what it binds. You can even express that protein and link it together with a small molecule. That's a piece of cake too. So, so if the tumor is heterogeneous and you have already 5 candidates, you can make 5 switch molecules. And then it should, CAR T are there for a long time. So if you're targeting multiple things at once, the chances are the tumor will be eliminated. But if it comes back, you can again do the biopsy a year later or something.
They are provided systematically, like orally or intravenously, or
CAR T therapy is given intravenously because all the biologics, they are not given orally. So depending on what kind of switch molecule you are giving. So if you're so it could be two small molecules linked together, it could be a protein linked to a small molecule. So depending upon the stability, you can decide. If it is, I would, yeah, if it is just a simple small molecule, maybe you can give it orally also. And it will be non-toxic because where will it go and bind? It is tumor specific, and one part is specific to CAR-T. Yeah, yeah. Go ahead. No, chemistry, it has nothing to do with the chemistry. So let's say that I am the switch molecule, right? So I am there. So there is one party here, one party here. Okay. So suppose I hold Shivani and Janvi like this, and I switch, then I hold Janvi and Venkatesh. Okay, and I do Venkatesh and Vanshika. So I am changing. I am the switch molecule and I am changing what I am holding. So I am the same, but my hands are different, holding different people here. So this is changing. I mean, humans can hold anyone. So this is not the best analogy, but basically, you can bring any two partners together. One is CAR-T partner, which I'm keeping constant because I have to get the CAR-T. So that ligand is always the same. So on one hand, I always have CAR-T. Here I can, if I change this, I can bind different partners. Okay? And what are these different partners? These are different tumor antigens. So I have the receptor for the tumor antigen, or I have the ligand for the tumor receptor. Make sense? No, 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 no. It's not like that. So CAR T cells are ready to go. Okay, they have the receptor, they have everything. So CAR T is just there. You don't change anything in CAR T. So CAR T is standing here, ready to go. And you know they have the receptor, this SCFP receptor. So I have to have the ligand to bring CAR T here. So this is the ligand which is holding onto CAR T. I'm the switch molecule, correct? I'm not changing CAR-T, I'm just holding on to that, okay, by non-covalent ligand interaction. So I have the ligand, CAR-T doesn't have the ligand, and then I have the ligand to bring the tumor cell. Or if tumor cell has a receptor, but if it is expressing some ligand, then I have the receptor there. Is it making sense? Yes, yes, absolutely. We are using T cells expressing uh, CAR T receptor, chimeric antigen receptor. Yeah, yeah. So you are right. CAR T is not standing on its own. It is part of the T cell because it has to use T cell machinery to spray, pepper spray to tumor cells. Yeah, you're right. That part is clear. So now you guys can see how awesome is that switch molecule. Tumor cells can keep changing their thing and you can keep changing the switch molecule. Okay. Just have that thing ready so that all of you can, you guys ask very good questions, should be uh, recorded. Go ahead.
Some CAR T cell have their life inside the body, so we are binding them to switch molecules outside the body, or we have once injected the CAR T cells and then we are switching the switch molecules.
We are, it can be given along with CAR T cells and it can be given right after the infusion of CAR T cells. The best thing is that to give them together. But once you give CAR T cells in the body, they are there for a long time. So you can also keep injecting these molecules to the patients. I don't know what is the half-life of these switch molecules, so maybe they need to be injected periodically to the patients.
Yes, they will be cleared.
They
from
will
the
be
body.
cleared, right? So you have to periodically inject that, but that is a piece of cake. CAR T is there. CAR T expressing T cells are there. Let me say it like that, right?
Ma'am, the affinity of the CAR T cells towards the switch molecules, when we find out that the first switch molecule we provided, the antigen for that is now not expressing and tumor is expressing other antigen.
Yes.
So we inject a new switch molecule.
Yeah.
So we will have to make sure that its affinity towards the T cell is more than the previous switch molecule. So the
So,
T cell will.
so if this is, let's say this is T cell and this is the T cell, this is the chimeric CAR, right? Chimeric antigen receptor, right? So this is going to remain the same, right? So the small molecule which will bind or whatever part which will bind will remain the same. We just have to change the top part which is binding to tumor cells in case it changes antigens.
But that one will be kept, matlab, it will be binded to the CAR T
Oh, I
receptor.
see.
So when we
No, so like Professor Venkatesh said, the small molecules will be cleared off by the body. So the older ones for which the tumor antigens have changed their thing, they will not stick around for that long, for years, right? T cells are there for years, so they have to be given periodically. So the old ones will be cleared and you can give the new ones, okay? But let me ask you, suppose they are expressing something which is not a receptor, how will you make something which will bind there? Anyone, can you tell me? So suppose the tumor is expressing some kind of weird hill-like structure, like some antigen on the surface. How will you recognize the tumor with that? If it is a receptor, it's okay. maybe you can make a molecule, but how will you recognize some kind of protrusion unique to the tumor cell?
Is it in the cell or just a callus, like the whole tumor?
It is a part of a cell. Tumor is just no, just the tumor shape will not do anything because that will change obviously. Like it is something sticking out of the cell.
Then how do we recognize it?
How will you make something in the switch molecule so that it recognizes it?
That time you said that antibodies can, that molecules can.
You can make a nanobody. Wow, guys, good. You can make a nanobody which will recognize the tumor. You have to get CAR-T next to it. See, now you have all the things in your arsenal. Just use, take out this, take out this. You already know that. You don't have to switch, keep a small molecule there. You can attach a nanobody there, right? To recognize it. All right, now we come to voltage logic gated CAR-Ts. Okay, add or not circuit. So what it means is that when it is a add circuit, so this CAR-T will recognize the tumor only when it binds two antigens A and B. Okay, if it binds A, it will not work. If it binds B, it will not work. If it recognizes both A and B, then it will work. Okay, so that will give high specificity. But again, the linker part is very important here, right? Or either A or B. Okay, now let's look at this. Not is sometimes the best one. So this is not A. So suppose there are two antigens, A and B. Okay, and I want you guys to think about it. So if it binds A, it will not work. If it binds B, it will work. If it binds both A and B, it will not work because A is not. So it can only bind B. So why it is extremely useful? So let me repeat it. There are two antigens A and B. Okay. So CAR-T will only work if it binds B. If it binds A, it will not work. And if it binds both A and B, then also it will not work because A is not. Okay. So why is that important? How can you use it? Why people have even made such kind of CAR-T? That's right. A is very good. A is also expressed in normal cells. So that is why if it is binding A also, then it will reject it. B is specific for tumor cells. Very good. That's why the NOT gates are very important. Okay. So this is here OR gate. OR means either A or B. Okay. So then it is more versatile. If tumor is heterogeneous, our thing is also heterogeneous. At least it can recognize two different antigens. So it binds A, it works. It binds B, it works. If it binds both A, B, it definitely works. And means alone A is not enough, alone B is not enough. Both together, it works. C is just the NOT gate. So when it binds A, it doesn't work. When it binds, when it binds A, it works. When it binds B, it does not work. When it is bound to both A and B, it still does not work. Okay, so, and this is because one of them is expressed in healthy, and that is the NOT signal. Okay, so I'm telling you, so this is very important for tumor heterogeneity because not one, if you're just counting on one, it may not be expressed or tumors may hide it. So that is why it is important to have the AND or NOT signal. Okay, and if you are checking two things together, they are likely to be more specific. So if it is a NOT signal, you are sparing the healthy cells. Okay, and it also works in immunosuppressive tumor microenvironment. Okay, so let's look at the AND gate again. It will only work if both A and B are working because it's AND. So it doesn't depend on A and B alone. It has to have work when A and B are together. So this is the OR gate. Either A, either B, both still works. If so let's go to NOT and AND. AND is, this will not work with bound to A alone, not work when bound to B alone. AB together it works. NOT gate, here B is the problem. So if it is bound to B, it will not work. If it is bound to B alone, it will not work. A and B are bound together, but since B is involved and B is also expressed in normal cells, it will not work. But only A, which is specific to tumor cells, it will work. So basically what they are trying to tell you that normal cells also express A to some extent, but they will also express B, but tumor cells will not express B. So you are making it extremely specific for tumor cells. So these are logic gated ones. If better, so here when it binds to A, it works both ways. When B is there in tumor, so tumor cells A, so that is great. If better gate, that is not commonly used, but here A is there, B is there, it is still working a little bit because A and B, because you still have A. But here B is completely absent, so this is normal, so it should not work here. Because, so problem is that A is expressed in all three of them. Okay, B is expressed in tumor cells. So this is a mixture of everything. So this is not a very good option because you will also kill the normal cells in there. So you have to make clear NOT signal. Okay. All right. OR gate means either A or B. Okay, so you already know this thing. NOT gate you already know. So NOT gates are also called as ICARs or inhibitory CARs. Okay, so ICAR detects antigen on healthy tissue, for example B. So if they recruit, if they see negative, if they see antigens on healthy, they said no, we are not going to attack it. So they bring inhibitory signal. When they detect tumor antigen, then it's a yes signal. Okay, so that is what it is. So it prevents toxicity to vital organs expressing low levels of tumor antigens. Okay, so this is a very good example. Antigen A, PSMA, is expressed in tumor cells. Healthy tissue express EpCAM. So, but healthy tissues can also express PSMA. So they will kill only when PSMA exists without EpCAM. So healthy tissues are completely spared, only tumor tissues die. So this is an example. This is inhibitory CAR, so it will not recognize the healthy antigen. This will say, okay, no, kill. So if both of them are active, then no one will win the game. If it says no, then no. Okay. When this is switched off, then only this will kill it. That's ICAR. Okay. So this is that inhibitory CAR. Okay, so now you can see why logic gating matters: off tumor toxicity and not improve specificity. When it is OR, then the advantage is that you have made against two tumor antigens. So at least it will bind one of them, so it will keep working. Okay, tumors are heterogeneous, so more A, B, C, D, E, F, G you put it there, better it is, right? But it has to be all tumor specific. So even if tumors are hiding the antigen, something will bind. Okay. So, and ICARs are very important to spare the healthy cells. Now let's look at armored CAR T cells. These are cytokine secreting T cells. So armored CAR T cells, what they do, so this is a CAR T cells. So once it recognizes the tumor, it actually releases a lot of cytokines and recruit other immune cells. So it is like, it is just saying, come, come, come. And the moment it says tumor cells, it will try to secrete cytokines. And different kinds of cytokines recruit different kinds of immune cells. Someone will recruit macrophages, someone will recruit NK cells. So basically, all these cells will come close to it and kill the tumor cells. So if the tumor cell environment is immunosuppressive, it will overcome that. Okay, because it is releasing the cytokines. So these are different kinds of cytokines which are released. IL-2, IL-12, when it is IL stands for interleukin. So IL-12, when it is secreted by CAR T cells, activates NK cells, macrophages, Th1 cells. IL-15 enhances CAR T persistence and memory. Okay, that is very good. You don't want CAR T cells, after spending $500,000, you don't want CAR T cells to disappear from the blood. You want them to be there. So IL-15 keeps them going. IL-18 converts cold tumors to hot. How would you define cold tumor versus hot tumor?
The tumor is susceptible to receive
Cold tumors are susceptible to receive they are resistant. They are resistant to receive tumors. Why? Why?
Aggressive,
They are
less attacking.
they are less
Attacking. They are more what you call inactive kind. Like
more than
they are
that.
not as rapidly dividing
So
as
you are
they
saying they are non-aggressive. That is not necessarily.
comparatively less.
They are most in a quiescent state of
It seems like cold just gives us an idea that they're quiescent, quiet, non-aggressive, but cold tumors are those tumors where immunosuppressive cells are absent. So, oh, sorry, not immunosuppressive. Immune cells are absent. So, all these T cell, B cells, NK cells, they are not there. So, they are in like real nice tumor suppressive environment, just enjoying their whatever it is. Okay, so if you have more immune cells infiltration, those tumors are hot tumors. Okay, and they are more, if you target them, they're more likely to be targeted because you have immune cells floating around. Cold tumors have very few immune cells floating around, not immunosuppressive, immune cells floating around. They are resistant to therapy, like she said, because there are no immune cells there. So cold cells are not necessarily non-aggressive ones. They could be aggressive ones, but they're also deeply buried in their own immunosuppressive environment. So nothing can reach them. Is it making sense? So they appear cold outside, but they can be pretty bad. But the thing is that if you think it in another way, if it is aggressive, it is going in all directions, then other things will also come to that. So in that respect, they won't be cold. But so that is why the definitions varies a little bit. But one way of looking at it is less infiltration of immune cells.
so ma'am, presence of only immune suppressive cell in their microenvironment makes them hot cells.
Immune suppressive. That is what I was saying wrong. When I meant immune cells, I was saying immune suppression cells. So say it again. What exactly do you mean? Immune cells or immunosuppressive cells? Cold are those which have immunosuppressive environment, and they don't have infiltration of immune cells like T cells or NK cells. Okay? All right. So this is what they are doing. These armored CAR T cells, they release cytokines and get the help from other immune cells. Okay, CAR NK cells are very important and they're becoming very popular. So we are always comparing CAR T cells with CAR NK cells. So CAR is the same. If you put it in NK cells, it is NK cell CAR therapy. You put them in T cells, it's CAR T. Okay. So CAR NK cells are engineered natural killer cells that express CAR similar to CAR T cells, but there are some differences there. So remember, NK belongs to innate immune system. And what is an innate immune system? From birth, and which is instantaneous. There is an attack, instantaneous. Okay, so that is innate. And naturally kill tumor or virus infected cells without prior sensitization is just immediate thing. Okay, so and we know that they originate from lymphoid cells. What are the cells? What are the immune cells which originate from lymphoid lineage? This is the kind of question you're going to get it in the exam. B cells, T cells, and NK cells. So lymphoid lineage is B cell, T cell, NK cells, correct? All right, so these are from lymphoid lineage. So look at this thing. So this is very interesting, the way NK cells recognize self versus non-self. So it is just really interesting. So remember I told you that every nucleated cell in our body has MSC1, and they keep showing their peptides to that. So NK cells actually recognize the MSC molecules. By the way, do you guys know what is HLA? Some of you are immunologists. So can you tell me what is HLA? Did you register yourself as immunologist? Otherwise, I'll direct it. You are immunologist, right? Okay, okay. So Shivani is going to answer. That's okay. Don't worry about it. If I'm put on spot, I'm blank too. Okay, so MSC1 in humans, MSC molecules, they're 1 and 2, right? Two classes. In humans, we don't call them MSC. Our MSCs are called HLA. They are identical. These are human leukocyte antigen, nothing but MSC. If you call MSC, okay, human MSC1, human MSC2. But humans have also given their own HLA name to it. So you cannot use HLA for a donkey or some other organism. They are MSC. All vertebrates are MSC, but we are the same MSC in us. It is called HLA. Okay, so here is the thing. Forget about the antigens. So NK cells recognize our HLA or MSCs. Okay, so they will. So my MSC is different from your MSC and your MSC and your MSC. So we all have unique MSC. So your NK cells will recognize your MSC and my NK cells will recognize my MSC. So MSCs, so every nucleated cell in the body have MSCs there. So NK cells are surveying and they recognize our MSC, and that's a switch off signal. That tells us it is self, right? But cancer cells, they are trying to be very smart, so they downregulate the MSC. So the MSC negative signal does not go to NK cells, they will kill it. Do you see that? Suppose, yeah, that is not a good analogy what I was going to say, but you get the point here. So if you downregulate MSC, NK cells will kill you. You're supposed to show up. Okay, so suppose you're coming through IIT gate, you better show up your picture. If you try to run, then they are going to follow you and put you somewhere else, right? So go through the proper surveillance process. So this is that signal here. So what happens is that this MSC, so this is NK cell, this is a healthy cell, and these things are MSC molecules. If it was human, we will call it HLA. So they are expressing, so NK cell says, oh, this is MSC. They say, oh, this is self signal. They will spare it. Now you make them disappear. These guys have made it disappear. So they said, oh, no MSC, foreign thing, kill it. So it is killed. That's how NK cells are surveying. Okay, so this is normal cell. They are showing MSC. NK cells find it and they said, okay, great, this is there. Okay, but if MSC has some foreign thing, then it will recognize that MSC is same, but the foreign particle is there. Then they will kill it because then they know there is some problem there. Tumor infected cell, they are trying to be smart. They take MSC inside. Now it will kill it. Okay, so that is the way they survey it. Okay, this is yet another one. So if they recognize that the MSCs are missing, they will spray it and they will kill it. Okay, so now if you make a CAR T cells near tumor cells, it's a very bad news for the tumor cell, right? It will just really kill it up. So you isolate T cells or NK cells. If you want to make NK CAR, you isolate NK, otherwise you isolate T. You insert the chimeric receptor gene, you expand them, you infuse it back, and now it is in the bloodstream. Okay, and now they will take care of the rest. Okay, now I'm sure you guys are very curious about what are the differences between CAR T and CAR NK cells. So there are some pros and cons on either side. Okay, so these are innate immune cells, you know, they spray perforin, granzyme, and all of these things and kill it. And this is minimal GVHD risk. So this is something I have written here. GVHD is graft versus host disease. So ideally it is very expensive to take the T cells or NK cells from the patient itself, right? And then engineer it and give it back to that. So suppose you take it from a healthy donor, right? So what are the things which are recognized by dendritic cells and all of these things, this HLA receptors? So if you remove all the HLA receptors, then it will not be recognized. But if you don't change it, then what happens when you give the T cell therapy or NK cell therapy from a healthy person to a patient? So this is a difference. So the cells which are coming will attack. So the cells which are coming from outside, the CAR T cell, they will think these are the cells inside are foreign, These guys will think you are foreign, they will kill each other. Cancer will survive, they will just kill each other. Okay? So when outside cells attack the patient thing, that is called GVHD. So for NK cells, GVHD risk is minimal because you are using NK cells which do not have a strong immune response against that one. But for CAR T, if it is allogenic, that means it is coming from a different patient, not from the same patient, then it is much higher. We have fixed that problem, but this is inherent problem. Okay, so allogenic works for NK cells, but for this you have to isolate it from the patient and put it back in the patient. But the good news is that this stay for very long time persistent. This has very short life. This can go for years. This can go for, 3-4 weeks to a month or something like that. Okay, so weeks, and this is sorry. Did I write the time here? I have not written, but NK cells don't stay for very long. CAR T cells stays for very long. This is CRS, ICANS. CRS is cytokine release syndrome and immune effector cell associated neurotoxicity. So if you give CAR T cells, body can rebel against it. Okay, and body can say, oh, foreign thing is coming. So these things are those things here. Let's not go in the detail. But for CAR T therapy, this kind of side effect is severe. NK cells, it is very low. GVHD is very rare for NK. If you give it from the patient to the same patient, no problem. If you take a healthy donor, it's a problem. See, ideally you would like to take it from healthy donor and just keep a lot in stock. And anyone needs it, you give it to the person. But then there is this problem. Allogenic use means someone else's thing. It's very limited. This is excellent. So all of these things, they have pros and cons. Okay. So limitations of CAR-NK, although safe and potentially cheaper, CAR NK cells face challenges, poor persistence. They don't stay in the blood for very long. Okay, lower in vivo expansion. They don't divide in the cell and expand. Once you give CAR T, they just keep on dividing. They are okay. And there is also problem in making CAR NK cells. The way the whole thing. Look at this thing, days and weeks versus months and years for CAR T. Okay, so what we are working on, researchers are working on improving NK therapy. So just to get you, I keep on saying allogenic and autologous. So autologous is when the patient's T cells are taken out and given back to that patient. That is autologous, no rejection. Allogenic is that you take it from healthy, you make tons of CAR T and then you give to patient. That is allogenic. So that is hard for CAR T cell therapy. Okay. Now let's go to the macrophages. We have made CARs in macrophages too. So macrophages are those immune cells which will go to the site, eat up the whole garbage and clean it up. So we have made CAR T cells, CAR M cells. So here you do exactly the same thing. You take macrophages and then you express the CAR receptor there. Okay. So the beauty of macrophages is that they are very motile. They run to the tumor site, they eat it up. So when you make macrophages CAR, they enter solid tumors easily. Okay. After this, remind me, I will tell you about oncolytic viruses also. Because yesterday we were discussing why oncolytic viruses are not so commonly used. So just remind me after this. So these macrophages, CARs, they enter solid tumors easily. They eat up the cell, they remodel the tumor microenvironment, and they activate the adaptive immunity. So they are really powerful, macrophages, CARs. These are one of the recent discoveries. Okay, and once they activate it, all of these cells are recruited. And once they are recruited, there is a strong immunity. So see, macrophages eat pathogen, and these are expressing chimeric antigen receptor. And then they eat up, they release these cytokines, all of these guys come in. And once they came in, these T cells are activated and they will kill the tumor cells. Okay, so these are CAR-M. turn cold tumors into hot tumors. So again, cold tumors don't have, you know, immune cell infiltration. So macrophages will go, and now they will recognize the tumor cell spelling is wrong. Okay, so cytokines are released, and then it will infiltrate the tumor cells. So this is mechanism of action. CAR-M cells are engineered. This is the CAR. They will target the tumor cells. So this receptor will target the tumor cells. So it binds there, and then it will eat up the tumor. It will eat up the cells in the tumor, and then it will secrete cytokines which will recruit all of these guys, and there will be enhanced tumor response, killing the tumor cells. Okay, so what are the advantages of CAR-M therapy? Better tumor infiltration, that is very important. Oncolytic virus cannot get into the tumor, that is the biggest problem. Multi-pronged anti-tumor mechanism, they're eating up the tumor, plus they're recruiting all the immune cells to kill the tumor. So it's a multi-pronged approach. This is CAR-M also have much more safety profile. You can imagine, these are going and eating up the tumor cells. They are not lysing it up and spreading all the garbage, they are just eating up the whole mess there. Okay, and this works for allogenic therapy also. Macrophages, you can put it in the body and there is not such a strong reaction. So they are supposed to have a broader therapeutic potential. So if you compare CAR-T with CAR-M, okay, so CAR-M, these things don't get into solid tumors. So there are many therapies which do not work very well for solid tumors because they cannot get inside the solid tumor pretty well. Oncolytic viruses are one of them. Oncolytic viruses only work for surface tumors like skin tumors and things like that. If you give oncolytic virus to patient, immune system clears up the virus completely. So there is no virus left to reach. Plus, even if it reaches a site, it cannot penetrate the tumors. That's why it has very limited efficacy despite being a very good idea. Okay. So dense stroma, dense extracellular matrix, macrophages, they eat up the ECM so they can get it in. Anything on the way, they eat it up. Okay. Immunosuppressive tumor microenvironment, you cannot stop macrophages from immunosuppressive tumor. They actually make the immunosuppressive thing to more immune responsive. So they will change it to hot. Low antigen density, no matter how low antigens are expressed, macrophages will eat it up. Okay, so CAR-M, and then they will recruit all of these immune cells. So they are very powerful. So CAR-T cells are just killing them. They are killing and eating them. Okay, then TME entry is poor. This is excellent. Antigen presentation weak. This is strong. This is best in blood cancer. CAR-T is exceptionally useful for blood cancer. These are very good for solid tumors, and they have more toxicity, they have less toxicity. So CAR-M is really becoming very powerful. Now let's go to the last one, which is universal CAR-T. So yesterday we were talking about CAR-T is so expensive, right? $500,000. It is personalized. So how we can make it universal? That we make it in a shelf, keep it in a pharmacy or in a hospital. Anyone who needs it, you give it to them. So if you have to make it universal, what is the thing you would like to do to a CAR-T? What is the problem? Why can't you give T cells from one patient to the other patient? Because of rejection, and which is due to MSC, which is called HLA in humans. So if you knock down HLAs, it should work. And then there is this TCR, which is T cell receptor, because CAR-T means T cells. And T cell receptors can also recognize, so you remove TCR and HLA, and you're good to go. And then you can make universal. Do you guys agree with me? That's what it is. Okay. So see, you are removing TCR, endogenous TCR. TCR is T cell receptor. So you're basically taking T cells, right? And then you're expressing CAR. So CAR is different because that's your creation. But it also has endogenous TCR, T cell receptor, right? you remove those. See, piece of cake to remove it. You just add the shRNA or CRISPR and you remove it. Okay, so we remove, and then we remove MSC1, which is HLA. Okay, and once you remove it, then these are universal CAR T. Why do we need universal CAR T? Because then you can take it from a healthy patient, make it and keep it and ready to go. Okay, so universal CAR T aims to create ready-made, standardized, rapid, accessible cell therapy. So how universal CAR therapy works? You remove endogenous TCR, you add the CAR, then you remove HLA molecules to prevent rejection. You add protection molecules. So you also add few other molecules. So if you add HLA, E, and G, then it will, this is very interesting. Explain to me what that means. Okay, so you remove some HLAs and then you add these HLA, E, and G to avoid NK mediated rejection. What does it mean? Can you explain to me? So how do NK cells recognize self versus foreign? By HLAs or MSC. So those MSCs which are recognized by NK, you put them back. So NK say, oh, this is self, right? Although it is foreign, but you're basically fooling NK cells as self, right? And you add all of these things, and with CRISPR, it is a piece of cake. Talents and this ZFN are also editing methods, not as popular as CRISPR, but all of these things are relatively much easier. So then it is ready to go. I could not resist. I wanted to give you some background about MSC and HLA because you guys have some diverse background. So I have added there. So MSC and MSC2 are the general name for these molecules in all vertebrates. This is just for your knowledge. In humans, they are called HLA. And these are MSC class 1 have A, B, C HLA. MSC class 2 have DP, DQ, DR. Okay. All HLA molecules are human. All HLA molecules are actually human MSC molecules. HLA is just the human nomenclature for MSC molecules. So this is what it is telling you, and this is telling you that MSC stands for major histocompatibility complex. It is present in all nucleated cell. MSC class 1. Class 2 are present in APC cells. Name one APC cell to be. Dendritic cells. And then, by the way, B cells are also APCs. Okay. Okay. So, and then HLA, you can see these are our HLAs. So key points, now you can understand the difference. When you call HLA, it becomes human, but they are actually MSC1. So this is all for your knowledge, that you don't get confused between MSC1, 2, and HLAs. So summary, HLA molecules are the main target of allo recognition. Our HLAs are different from each other, okay? So if you remove them, there is nothing there. So knocking them out reduces immune-mediated complications in allogenic therapy. This is the key strategy for universal CAR T therapy. Knock out HLA, hide donor cells from host immune system. Okay? All right. This is just everything I have told you, just very complicated way, but it is all listed here. We have discussed every single thing. Okay? So now you know what is the best way to do it. You make CAR T, you remove TCR, you remove the HLA which is causing rejection, and then you can just keep it in the freezer and infuse it as needed. So this can become a lot cheaper, okay? Now we come to, so the title of this was NK therapy and dendritic. So we have to finish with dendritic cell therapy in the next couple of minutes. So dendritic cells, you guys know, these are APC cells. So dendritic cell therapy is a type of immunotherapy that uses patients' own dendritic cells to train their immune system to fight diseases like cancer or autoimmune diseases. So you isolate dendritic cells, and then what do dendritic cells do? They recognize the antigen. So you take them out, and outside you train them to recognize the antigen. And once they are really primed, you put it back in the patient, and so that they can go and kill the tumor cells. So what they are doing is that dendritic cells recognize the antigen. So PAMP and DAMP, these are, PAMPs are pathogen released. I've written what they are called. These are pathogen released antigen. DAMPs are damaged, our damaged cell released antigens. So they are recognized by dendritic cells. they release cytokine, recruit all immune cells, or they, they are APC cells, dendritic cells, so they will present in MSC. TCR will recognize it, and then T cell will be activated, and B cells will be activated, and either T cells will kill it, or they will activate B cells to release antibodies to clean up uh, the the foreign particle. So this is dendritic cell. So when it recognizes viral infection, it gets activated. Once it is activated, now this is very interesting. The dendritic cells will take it to the lymph node. Why it will take it to the lymph node? Because B and T cells live in lymph nodes. Okay, so suppose two really great people, like two very powerful group of people live there. So T cells live in T cell zone, B cells live in B cell zone, and they have a boundary. So what will happen is that this will these dendritic cells will go to the T cell area, and then they will present it to T cells, and T cell will get activated. So then these T cells who are activated, now they will migrate towards B cells, but they will not go inside B cell, they will wait in the boundary. At the same time, what happens, so B cells, they recognize the three-dimensional structure of the virus. They will not take the chewed up peptides. So they need the whole thing. So there are another kind of dendritic cells which are called follicular dendritic cells. They will present the whole thing to B cells by going to B zone in the lymph nodes. So B cells will recognize this. They will take it inside. They will chew it up. Now they are ready to make the antibody, but they need a second signal from the T cells. So now they will go towards T cells. T cells are going towards B cell. So where T and B cell boundaries there, they will meet. So B cells will present the chewed up stuff to them. T cells are already primed because they have received signal from dendritic cells. So T cells say, oh, okay, they will lock on and they say, you're good to go now. And then B cells will mature to plasma cells and release antibodies. Is it making sense? Should I repeat the whole thing? Maybe one more time, quickly. Okay, so what happens is that dendritic cells sense viral infection. Okay, then they take this, chew it up, and then dendritic cells will go to the lymph nodes. Lymph nodes are where B and T cells reside. So let's say half is B cell, which is B cell as a T cell zone, half of it. It's not half and half, but I'm making it easy, and this is B cell zone. B cells live in their zone, T cells live in their zone. So dendritic cells don't go to B cell, they go to T cells, and they will present, they are APCs, so they will present the chewed up stuff there. T cells will get activated. They will come to the meeting point for B cells. Here B cells independently got the virus, which was presented to them by another class of cells. So they now take the virus in, they chew it up, and they know that there is something wrong. But as a fail-safe mechanism, they have to get permission from T cells. So T cells are already primed, B cells chewed up, they have taken the virus, chewed up, they come and meet T cells. They both connect together, like actual connect together. T cells say, good to go. That's when B cells will become plasma cells, and then they will secrete antibodies towards that antigen. Make sense? That is the mechanism. Okay? So this is more complicated than what I told you. See, AI cannot make a perfect thing. I was struggling, I was feeding thing. Then I realized I can tell you better and you can figure this part out. Some part is correct, some part is a little bit here and there. Okay? If it was Purdue, I would make it myself with all the tools I had there. But here I was waiting for that. Okay? So this is just telling you that you can also make differentiated dendritic cells. So how do we do dendritic cell therapy? I got sidetracked. I was just telling you how antibodies are made, but let's come back to dendritic cell therapy. We take monocytes. Monocytes can be converted back to dendritic cells. So you give them these cytokines. They are mature. Now you expose them to tumor antigens. Now they are ready for those tumor. Now you put them in lymph nodes. They activate T cells. And then once the T cells are activated, they will kill the tumors. Okay? So that's the whole process here. All right? This is yet another picture of showing how they go and talk to this V zone and T zone. All right. Tea time. Okay. So we'll come here at in 15 minutes. So what will be? 10:50. Okay. Can I answer it after that? Yeah, I will answer all the questions. Okay, let's start now. So we start with something we all want to learn now. So after hearing about cancer for so long, we want to prevent cancer, right? So that's where we come to immunoprevention. You are using the immune system to prevent cancer from happening. So you all saw how important is immune system. Cancers can survive because they close our immune system. So if we keep our immune system active, we can actually prevent cancer. So this is showing you immunotherapy strategies. You guys know about PD-1, PD-L1, CAR-T, CAR-M therapy, cancer vaccines, monoclonal antibodies. You can also use these to prevent cancer. So immunoprevention approaches, the best one is prophylactic cancer vaccines. You take cancer vaccines in advance, right? Some of you have been giving me ideas about vaccines, and as I'm putting together the lectures, actually your ideas have been realized here. So even if they are realized here, you came up with this idea, so they are original ideas. Somebody just did it. So that should give you a lot of confidence that you're thinking in the right direction, right? Because you're just learning about it, and that's how you're suggesting new ideas. So cancer vaccines are available for many different kinds of cancers, particularly those which are viral infected cancers. Okay, then surprisingly, inflammation is a very important thing for cancer. So these headache medicines that you take, aspirin, like that is also cancer preventive, because you are killing inflammation. So the root cause is that you should never have an inflamed tissue for very long. Okay, that's very bad news. And there are other anti-inflammatory therapies. So we know what is cancer immunotherapy. We know you harness the immune system to detect and eliminate cancer, because tumor upregulates immune evasive mechanisms. And you know about PD-L1, CTLA4 therapy. You know about CAR-T, CAR-NK, CAR-M therapies, cancer vaccine, cytokines. So immunoprevention is preventing cancer by priming the immune system before disease comes. Okay, so includes vaccine against oncogenic viruses. So especially if you are going to visit some countries where there is a chance of getting infection, right? So your immunity is very high being in India, but when people come from outside, and if they're exposed to something for which we have natural immunity, then they may get a disease, not necessarily cancer or something, but so if we many times we take vaccine before we go out elsewhere, right? This is to prevent a disease before it happens. Okay, so reducing the risk of cancer. So you can take vaccine. So it is very common to take HPV vaccine against cervical cancer, Hep B vaccine against liver cancer. So liver cancer can occur due to Hep B, Hep C. So if you take the vaccine, you can really prevent that disease. It's just very effective. Chemo prevention. You guys know tamoxifen is a breast cancer drug. So people who are prone to have breast cancer, they have a history of breast cancer, they take tamoxifen in advance so that they don't get it. Look at that, aspirin is one of them. Okay, so and then people who have BRCA1, BRCA2 mutation. Please remind me, Venkatesh, you asked me about BRCA1, BRCA2 and radiation and olaparib therapy. So we'll discuss that also today. Remember you were saying, so whenever olaparib therapy is given, it is followed by chemo, it is always accompanied by chemotherapy and radiation therapy. Chemo or radiation therapy, because you have to damage the DNA. But I found even when it is germline mutation of BRCA1, BRCA2, usually one allele is there. But in tumor, both alleles are lost. Both copies are lost, but the human being has still one allele. So that's why it is more damaging to the tumor cells. Okay, so if somebody has BRCA1, BRCA2 mutation, you either remove the breast completely, that surgery is called mastectomy, or you remove the ovary, that is called oophorectomy. Okay, so like Angelina Jolie, she got rid of the ovaries, fallopian tubes, as well as the breast, because she was at very high risk of these cancers. And then the lifestyle intervention plays a very important role. No smoking, limit alcohol, healthy diet, regular exercise is the biggest chemo prevention for cancer. So this is the lifestyle thing I'm writing. So maintain a healthy weight, eat vegetables and fruits, be physically active. Okay. If mothers are breastfeeding their children, it reduces the breast cancer chance tremendously. Fast foods are very bad, like McDonald kind of food. It's a joke, like McDonald kind of food. French fries and all of these things are very bad for your health. Red meat is very bad. Alcoholic drinks can lead to liver cancer. Okay, and Coke, Coca-Cola, or Sprite, or any of those drinks, they are loaded with sugar. They are just pure sugar. If I go to my dentist, there's a picture, instead of soda, you are actually eating sugar directly. It's that much sugar. So it is extremely bad. It's just like sugar hidden in the form of cola. Okay, so you have to limit that thing. So this is just showing you how cancer progresses. So these are healthy cells. The cancer, one cell has initiated, now it is forming a hyperplasia. These are different stages. Okay, now cancer has properly formed. Now it is becoming invasive. It is going, and now it is metastasizing to the bloodstream, and then it goes and lodges somewhere. So immunoprevention can be done before cancer forms or at the early stages of cancer. Once it becomes metastatic, it is metastatic. Then you are just doing therapy. There is no prevention there. But you can prevent it from progressing further. Okay? So based on that, there are three kinds of immunoprevention: primary, secondary, and tertiary. So primary is that prevent disease in healthy individuals. So people who get surgery done, ovaries removed, that is preventive surgery. They are just high risk of cancer, but they don't have cancer as yet. So that is preventive. Okay. Secondary is that they have precancerous lesions. So they have small lesions, like little bit of this kind of thing, but they are just really taking proactive thing. Many times people go for some kind of test and they have some kind of polyps in the rectum or in the intestine, and so these are just benign lesions, but they're proactively taking care of that. That is also prevention. Tertiary is that, the person had cancer, but now they have gotten rid of cancer, but they are taking preventive therapies so that the cancer does not come back. Okay, so that's what it is. Primary, healthy individuals, but at risk. Who are the people who are at risk? BRCA1, BRCA2 mutations. People who are smoking, or people who are not smoking but working in bars where other people are smoking. So they are at high risk of cancer. Okay, so and then these are some diseases. Lynch syndrome, these people have, if they have Lynch syndrome, they have high risk of cancer. Okay, and once the cancer is developed, like colon polyps, if they are there, it can go to cancer. So this can be treated at this stage. Okay, so this is so so viral infected cancers are very easy to treat for prevention, not treat for prevention, because we know the viruses cause that. So we have vaccines for those. And if we give the vaccine, the person is saved, right? That person may not develop cancer. So for this HPV virus, it gives rise to liver cancer. So we have different vaccines. So these are given to people who do not have cancer. HPV1 VP, it is approved. You give the vaccine, and even if HPV virus attacks, they already have the immunity against it. So this will not develop into cancer. But at lower stage also, we have a vaccine which can be given. This is slightly different vaccine, you can see. So this will recruit all the immune cells because it's a vaccine, and then we can treat it at this stage. For high grade, also there is a vaccine, it's a cancer vaccine, right? We know about the cancer vaccine used for therapy. But this is the preventive one. These are the treatment ones, okay? So immune system plays a very important role, and that is why immunoprevention is very important. So you guys already know about innate immunity and adaptive immunity. T and B cells are the ones which come in the adaptive, right? They make the antibodies. B cells become plasma cells, secrete antibodies. Okay, so these are the adaptive ones. These are the innate immunity ones, which we have already discussed. So biomarkers play a very important role. How do you know these people are at high risk, right? Some people are healthy, they still take vaccines, especially if they're going somewhere where there is a chance of getting infected, they will take vaccine in advance, right? So people who are doing, uh, they go in jungles or they do, they do archaeological kind of work. So you don't know what kind of things you are going to get exposed to. So they are preactively taking all these vaccines. So traditional approach, this is the traditional approach. You have high risk patients, you have two sets of high risk patients, but treatment works for them, it doesn't work for them, and that is determined by the biomarker. Okay, and you already know about the precision medicine approach where the biomarker testing is done. So I'm going to tell you about 6 different ways by which we can prevent, we can harness the immune system, because this is not prevention of cancer, this is immunoprevention, that we are harnessing the immune system to prevent cancer from happening. So cancer vaccines, and we already know about this one, you have seen this picture. There are many kinds of vaccines which you can make, both as therapeutic after cancer occurs or preventative vaccines. So look at these viruses. These are well-established viruses which lead to cancer. So HPV leads to cervical cancer. EB virus gives Hodgkin lymphoma. Human T lymphotrophic virus gives to T cell leukemia. This virus to Kaposi sarcoma. Hep C, Hep B, they give to SCC, hepatocellular carcinoma. Okay. So now we know about this thing. So about 30 to 50% of all cancer cases are preventable. That's good news. 50% of cancers can be prevented. So cancer-causing infections such as Hep B or HPV are responsible up to 25% of cancers in low and middle income countries. Vaccines are available for this and sometimes of HPV. So if you give it to them, you can prevent the disease from occurring. So that's really good news, right? So so what happens when you give vaccine? You guys already know it, but I still want to go through this once again. So when you give vaccine, right, you are taking tiniest antigen of that virus, right, which is not going to do anything except trigger an immune response in your body. When it triggers immune response, B cells will form antibodies, right? So when B cells, so first T cells will get activated, then B cells will get activated, and then they will provide you with the antibodies, right? They provide you with the antibodies. But if they just give you antibody one time, it is no good. So then what they will do, some of these antibodies will become memory antibodies and they will be stored for long-term storage, right? They will change the kind of antibody. So the epitope will remain the same, but now you prepare a different kind of antibody, which is a long-term antibody that will be stored in our body. So initially you will make rapid for the immediate clearing of virus, and then you save some of those for long term. So anytime that virus infects again, you already have the memory antibody and they will rapidly proliferate and very quickly they can attack the things. So these are the memory ones, okay, which are stored here, okay, and these are rapidly stored. So that's how immune response works. So what is memory immune response? So after when the adaptive system encounters an antigen, after the initial response, a fraction of these cells becomes memory cells which remain in the body for years and decades. Okay. So these memory cells can respond faster, stronger, more specifically than the first encounter. So when we were kids, like when we were born 2-3 months old or 15 days old, we got several vaccinations, one after the other, and we still have that immunity within us because of these memory T cells. Okay, so cancer immunoprevention aims to prevent cancer before it occurs by training the immune system to recognize and eliminate precancerous cells or oncogenic viruses. Okay, so virus like HPV virus or HPV virus. Okay, so HPV vaccine prevents cervical, anal, head, and neck cancer. So many of them. HBV prevents liver cancer. Liver cancer is pretty bad. Liver is a very important organ. Okay, so these vaccines create immune memory. So if the virus appears later, our body is ready to fight it off. Okay, neoantigen based or tumor antigen vaccines. So some cancers develop from known mutations like for KRAS, G12C, G12D, all of those mutations. So we have made vaccine against them too. Okay, and if you give that vaccine, if that comes in, then those B cells, those antibodies can clean up those tumors too. Okay, so memory, so we have also made against these guys, EGFR receptor, it is expressed in which cancer? Lung cancer, very good, and breast cancer. Okay, so vaccine can expose the immune system to these antigens early, creating that CD4, CD8 T cells, and they will. So people who have Lynch syndrome, BRCA1, BRCA2 mutation, strong family history, chronic inflammation, these people are extremely high risk for cancer. So tumor, so there are two kinds of antigens, tumor associated antigens and tumor specific antigens. So tumor specific antigens are neoantigens, which are only expressed in tumor cells. And tumor associated antigens can be expressed at low level in normal cells also. So ideally you want to go for tumor specific antigens to have a very strong response specifically against the tumor. Okay, so this is just telling you that when you give vaccine against, or you make against a new antigen, which is a tumor specific antigen, then you will spare the normal cells and you will kill the tumor cells. Okay, so cytotoxic T cells will find those antigens and they will kill the tumor cells, and they will recruit other B cells, will make the antibodies, and antibodies will bind and kill the tumor cells. Okay, so this is the difference between tumor specific antigen and tumor associated antigen. Tumor specific antigen, as the name suggests, is present only on tumor cells. These are TA are present in normal cells also, but highly expressed in tumor cells. So normal cells have small amount, tumor cells have a lot more. These are usually mutation of like R12C mutation in RAS, KRAS. So that becomes tumor specific, because anytime you have G12C or G12V or G12 mutations, that will give rise to cancer. So that is a tumor specific one. Okay. Uh, so this is mutant P53 RAS viral proteins, and they have a very strong immune response. If you make a vaccine, there will be a strong immune response. T cells, B cells, all of them will get activated. So TCA is tumor exclusive, tumor specific, better target for immunotherapy. TAA is also expressed in normal cells, although in small amounts. So this is, you don't want to use these when you're making immunotherapy. So new antigen vaccines are good because new antigens are specific for tumor cells. So what you do, first you have to find the new antigen. So you do the sequencing and then you will make a peptide. See, this is a tricky part, okay? So suppose even if you know KRAS mutation is there, what part of KRAS would you make? So these programs can detect if you make this sequence, that will be highly specific. And then based on this, you make a vaccine, then you inject it, and then the body will take care of the rest. Okay. You guys already know about immunoprevention by immune checkpoint modulation. So you give PD-L1 and PD-1 therapy in advance. The person who is likely to develop. Tell me a case when a person is likely to develop, but he or she has not inherited from the parents. Who can be the other group of people who are highly likely to develop cancer? Exposure to chemicals, exposure to smoke, exposures to radiation. So these people, or exposure to some kind of viral infections. Suppose you are working in a hospital with the patients who are infected. I mean, you have to take all the precautions to keep yourself safe. Okay. So you already know about the checkpoint modulation, CTLA-1/4 therapy, those kind of things here. CTLA-4 here, right? PD-1, PD-L1. Okay, so immunoprevention by checkpoint modulation restore T cell surveillance because T cells are shut off by PD-1, PD-L1. So if you take that therapy, they are always active. You have memory T cells, remove early transformed cells. Transform means cancerous cells. The cells which are at the early stage, if you remove them early on, they will not form the tumor and halt progression of precancer, and you get long-term immunity. Okay, so you target high risk population. This strategy focuses on individuals, of course, with a genetic risk of developing specific cancers. BRCA1, BRCA2, P53 mutations, all of these things. Okay, so preclinical evidence has shown that in mouse models, prophylactic, which means preventive immune checkpoint blockade, can significantly delay tumor formation. So one can, if the person is at high risk, they can preventively take PD-L1, PD-1, CTLA-4 therapy. The body is healthy, they can deal with the side effects also of the immunotherapy. Okay. All right, so this is, you already know this very well, PD-1, PD-L1, so I'm not going to tell you about that. Okay, so mechanism of cancer immunoprevention by checkpoint modulation. So T cells get exhausted. Why? Because they become quiescent, because PD-1, PD-L1 get locked and T cells shut down. So if you do the PD-L1 therapy, T cells are re-energized. So when T cells are energized, if there is anything wrong, any precancerous cell, they will be removed and they will not form the tumor. Okay, then B cells will be active against them. They will make the antibodies, clean up the whole system. Then they will keep the memory cells, so the tumor can never form again. Okay, so and breaking immunosuppressive environment, so you can clear the immunosuppressive environment. So that is a very good way to keep cancer at bay. Okay, so now let's come to, this is what you were saying, Shivani, right? Monoclonal antibodies in immunoprevention. So that's where they come into picture. So you guys know how monoclonal antibodies work. What's the difference between monoclonal and polyclonal antibody? Can you tell me? Anyone? How do you define an antibody? It is a Y-shaped molecule, but what does it do? It binds to the target. And the part where it binds, what is it called? Epitope. Or epitope, it's called epitope, it binds to this epitope. So polyclonal, this is a protein. Polyclonal antibodies are many different species of the same antibody, which is recognizing different part of the same thing. But monoclonal will always bind to the same epitope. It's against only that one epitope. Polyclonal are just attacking many epitopes. Make sense? So this is a protein. So if it has polyclonal, it has different species. It will bind here, 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 here. So it's a mixture of antibodies, but they're all targeting the same protein, but binding a different site. So it's a mixture of antibodies. Polyclonal will just bind at one site. So, monoclonal will just bind at one site, and that only one site. So every species will only bind at that one site. So that is monoclonal. Okay. So that's what it shows, monoclonal. It is showing you like this, but they are identical. These things are identical. Okay. And once attached, either that whole thing will not work, or it will trigger immune response and all other cells will come and eat it up. Okay, so this is a monoclonal antibody. They're binding to the same antigen. Okay, and once the monoclonal antibody binds, this causes the immune system to attack the cancer cell. It's like this is HER2 receptor. When Herceptin binds, then immune system gets activated and it comes all this killer cells will come and kill the tumor cells. Okay, so there are monoclonal antibodies in immunoprevention also. So we have antibodies against HPV, EBV, experimental antibodies, which can be given preventatively to prevent virus-induced cancers. Checkpoint blockade antibodies can also be given when the cancer is just developing, or if the person has high chance of developing a cancer. Okay, these HER2 antibodies, if there is a family history of that, you can give it in advance before the cancer develops. Okay, anti-inflammatory antibodies, there are many inflammation driven cancer. We know inflammation gives rise to cancer, so you can give anti-inflammatory therapy in advance. You can give aspirin. I mean, aspirin is just very small thing, but you can give stronger prescription medicines for anti-inflammation in advance. That is also preventative thing for cancer. Okay, if it is just becoming precancerous, you can give ADCC, killing of early clones. Remember, antibody binds and the whole immune system comes and attacks it. So that's what you can do early on, before cancer is actually there. Okay, so there are many ways to target it. Okay, you can give antibodies bind to radioactivity. Basically, all the things you give it in therapy, you can preemptively give to the patient to prevent the therapy, to prevent it. Except, when the cancer is there, you don't give aspirin to the patient, because by that time aspirin is not going to work. But there are many other things which will still work. Okay, so that's the whole thing. You can give immunotoxin, you can give cytokines, you can give radioactivity. Okay, so that's all about monoclonal antibodies in immunoprevention. Now let's come to adjuvants in immunoprevention. So what is adjuvant therapy? Can you remind me? After the main thing. Okay, so you can actually give adjuvants in immunoprevention. I'll tell you what it means is that, so when you give preventive vaccine many times, it does not, so oftentimes the neoantigen may not be very immunogenic. Okay, so suppose there is a protein, right? So this part will trigger immune response, but this part will not trigger immune response. So some parts are recognized by immune system, like really they will respond to that part, but they will not respond to some other part. So this part is weakly immunogenic, this part is strongly immunogenic. Okay, so if your neoantigen is weak, then it is not going to work. Or suppose you're even giving a vaccine. Why are you giving a vaccine? To turn on the immune response. And if the vaccine is not turning on the immune response, it is useless. You are adding oncolytic virus, but the virus itself is cleaned up by the immune system, right? You want vaccine to go there and body to really strongly react and trigger immune response. So sometimes you add adjuvants. These are just some compounds that you add. When you add them together with the vaccine, the immune system reacts very strongly. That's all it is. So you give very little vaccine, but you mix it with some small molecules, and the immune system will strongly respond and make antibodies against it. That's all it is. So preventive vaccine without adjuvant often has weak immune response, low efficacy, but if you mix it with adjuvant, strong, durable, targeted immunity, and you can prevent this. So what these are adjuvants and substances added to vaccines to enhance the body's immune response against a specific disease, which makes the vaccines more effective. Okay, adjuvants are critical in immunoprevention because preventive vaccines often target self-like tumor antigens which are weakly immunogenic. So if you add adjuvants, they wake up the immune system to recognize even when the tumor is full blown, it is easy to recognize. But you are doing preventive here. So preventive may be very little tumor cells, but so that's when immune system has to be super vigilant to find whatever is hiding there. So that's why adjuvant addition helps in vaccines. Okay, so vaccines and adjuvants, they alleviate the problem of limited vaccine supply, because now you have to use limited vaccine, because small amount of vaccine will work because you have added the adjuvant. enabling a more rapid immune response, antibody, increase the magnitude, developing vaccine for effective T cell. Okay, so this is much better. All right, we know chronic inflammation is bad. Okay, so so whenever there is inflammation, there is a risk to develop cancer. Chronic, not just simple inflammation. So blocking pro-inflammatory cytokines. So there are cytokines which are released when there is inflammation. If you clean it up, they cannot damage it. Okay. Then inflammation takes the immune cells and make them immunosuppressive. So which are supposed to clean up the tumor cells, they are the one which are promoting tumor. So if you clean up that, you can add compounds. Remember I told you about the STING pathway. So if you give the activators, you can prevent the cells from becoming immunosuppressive. Okay, so you are preemptively trying to make you are preventing the tumor to make a tumor suppressive environment. You are giving the drugs so that there is immune system is fully active. Okay, neutrophils can lead to inflammation. You can target that. You can obviously use PD-L1 therapy because PD-L1 therapy is waking up the immune system. Okay, and microbiome also, tobacco smoke alters the lung microbiome. So you have to really make sure that in such case, which promotes inflammation. So microbiome modulation is also an emerging area. Okay, and oftentimes we, use combination therapy, vaccine with PD-L1 therapy or vaccine with anti-inflammatory drugs. Okay, all right. Prophylactic surgery. So the most common one is that bilateral mastectomy. You remove both breasts. Okay, these happens with BRCA1, BRCA2 mutations. This surgery is to remove ovaries and fallopian tubes. People with BRCA1, BRCA2, high chance of cancer. So you remove the ovaries. See, they appear pretty simple to do that. In some people, there is no side effect. But I know of a lady who had both of her breasts removed and she's in the hospital every 2 months for a month. So she will be in the hospital for 1 month. For 2 months later, she's back in the hospital because they just messed it up. So there is some infection or the other. So 4 months every year she spends in the hospital. Okay, so so surgery also comes with its own risk. Okay, especially if the doctor is not very good. Okay, you guys may have heard of thyroid gland removal, right? How many of you have heard of that? All of you. So let me tell you a bad story about this thing too. So, like levothyroxine we take for thyroid. So I had a colleague in my department in chemistry. So she had hypo or hyperthyroid something. So she was totally fine, no health issue in her 40s. And they told her to get this irradiated so that it is not working. And they said you have to take levothyroxine for the rest of your life, and that's it. So she was joking that I will buy this big bottle of levothyroxine because I have to take it for life. So she was planning her surgery on November and December. So I said, I can take all the classes while you are in the hospital. So I have no problem in taking the classes in your absence, her classes. So she said, no, don't worry about it. I have made the plan. Students will present papers during that time. So you don't have to worry. Completely healthy lady, just like no problem, absolutely no problem. They did the irradiation there, and the irradiation went for whatever time that caused cancer. X-ray, radiation can cause cancer. 6 months later, she was dead. So basically, the doctors killed a completely healthy woman. Okay, so one has to be cautious when they are doing the preventive thing too, because that can be uh, very bad. So if the doctors are telling you something, you guys are scientists, read about it. Okay, don't let the doctor drive your wagon. Okay, all right, so I'm quickly going to give you some cases. Go ahead. Mm-hmm.
shows more percentage of breast and ovarian cancer compared to other ones. Like it shows the DNA repair mechanism.
So BRCA1, BRCA2 also is expressed in many other cancers. Prostate cancer, a high percentage of BRCA1, BRCA2 is there. So, but it is not present in all other cancers. So it is possible that those other cancers have other repair mechanisms. They upregulate other repair mechanisms. So it could be some pathway which is still intact in those cancers. And breast cancer and ovarian cancer and prostate cancer really depends on BRCA1, BRCA2 pathway. So that may be more susceptible to it. Because it makes sense, if they have BRCA1, BRCA2, they should be prone to every cancer. But these cancers are more susceptible. There is another way to answer that. BRCA1, BRCA2. So remember I just told you, so yesterday I checked. So even if there is BRCA1, BRCA2 germline mutation, every cell of the body, it's one allele only, not double allele. Okay. So in breast cancer, prostate cancer, ovarian cancer, and one other cancer, in this both BRCA genes are deleted. So it is these cancers which have both BRCA genes deleted, both copies of the genes deleted. So that's why they are more susceptible. I did not see any case yesterday where there was a germline mutation of both of them deleted. So is it making sense? So even if it is germline, you still have one copy of that. So they are still able to repair genes, but the tumor itself does not have a single copy. And these cancers, these two copies are deleted. Make sense? All right, so now I'm going to tell you some success stories using immunoprevention, right? So this is a real case about HPV vaccine. So if you give HPV vaccine, you prevent cervical cancer, okay? So in Sweden, when this vaccination was given to girls less than 17 years, 90% reduction in cervical cancer, okay? In Denmark, 86% reduction, huge reduction, right? US, 12 to 22%. It is possible that, it is not that prevalent there. I don't know why it is less in US, okay? But just see how much prevention is there. Okay, colorectal cancer, a deemed coming true. So some cancers are highly aggressive cancer and they develop very quickly. They are there and then they are not there, and all of a sudden 1 month later they are full-blown cancer. This is one of the slowest growing cancer, colorectal cancer. It may take 10 years to develop, like really slow. So the good news is that one can catch it at an early stage, and when you catch it at the early stage, you can fix it. So immunoprevention therapies work extremely well in colorectal cancer, okay? So there are drugs and you can catch them at early stage and you can prevent this cancer, okay? Then breast cancer, HER2 immunopreventions. So if someone has family history and it is likely that they have HER2, then you can prevent it before it actually develops into a cancer, okay? High-risk women, family history, and BRCA mutations may benefit from preventive strategy, including HER2. So there are different kinds of vaccine against HER2. It's a very famous target, right? So vaccination before the cancer onset in genetically susceptible women, you can give immune therapy in advance to really boost up the whole immune response there. Liver cancer, like HBV virus, leads Hep B virus, Hep C virus. So you can give vaccine in advance, liver cancer will not develop. So that's a very good one. Okay, lung cancer, obviously whenever there is lung cancer, smoking comes into picture. So lung cancer remains the leading cause of cancer death worldwide, largely driven by tobacco exposure, chronic inflammation. Okay, so immunoprevention. So if you can inhibit chronic inflammation, you reduce the chance of cancer. And then because lung cancer is so prevalent, they know a lot of neoantigens. So you can make vaccines against neoantigens and you can give it in advance. and you can prevent that disease. Okay, so that is another way to reduce that. So immunoprevention is proactive. Vaccines, antibodies, immune modulations are key. So remember, it is immunoprevention, not prevention. If it was just prevention of cancer, I would say eat healthy, exercise, don't drink alcohol, all of those things. Okay, that's it. Okay, so it's 11:30. So we'll wait for 5 minutes, and 11:35 we'll start another lecture. So we'll at least finish one lecture before lunch, and if we finish one lecture, we'll start the next lecture, and then after lunch we'll what time is lunch today? Regular time, 1 o'clock. It is at 1 o'clock. So let's. You guys can ask me question, and if you want to take a break 5 minutes, you're welcome to take the break also. Huh? Yes, go ahead.
Does vaccine have something to do with age?
Uh, you have to use that book.
Ma'am, my question too is, does vaccines have something to do with ages? Because if it is, then when we are given administered with human papillomavirus disease vaccines, there are certain designed towards the age limit. Like there are certain different dose to women who are, females which are under 5 age, then to 14, and then to post 20s. Is it something related to
I
that?
don't know about that. The dosage is changed according to age, but if you say so, I can accept it, because if you give it to older people, you don't want to give them a very strong dose, because their body may not be able to handle it. Younger people, so they can change the dose. But how does it matter even if they change the dose? So what is your question?
Does antibody effectiveness gets damaged, or does it have something to do with age? That's the question.
No. I don't think so. Usually, what happens is that younger people are more resilient to everything. So, let me tell you something. Okay. Um, is it on? It is on. Okay. So, what happens is that if you take out the neurons from young mice, brain neurons, okay, and you give them insult, you have to give them huge insults before they die. And if you take the same neurons out of the from older mice brains, older mice means 3 months old, they are old. You can give little bit of insult and they die. If you take, so for some time ago it was allowed in US, so to take the abort fetuses, human fetuses, and you can isolate the brain neuron cells from there. So my collaborator, he used to do that. So in Alzheimer's disease, you add amyloid peptide, which is an insult. So if you add it, then basically neurons die from all adult. But if you give the same thing to fetus's brain, right, they proliferate. So something where they should die, they proliferate. So the reason I'm giving you these examples are that younger babies, obviously you have to look at the dose and everything, but they are very resilient in many ways. They are better than adults in some ways. So I'm sure that they do the dose thing because it's a baby versus an adult body. They do that. But I would think that the immune response would be equally effective whether it's a baby or an old adult person. Older people, maybe it will be compromised a little bit because, the whole body is shutting down basically, right?
Okay. So basically, the ideology was, if it has something to do with age, I'm talking about individual. I'm considering an individual B cell, for example. Does it gets denatured, or does something get
No, no, no,
denatured
no.
according to age?
No. If it is denatured, it will be eliminated. It will be eliminated in a
Even
new
if
B cell.
single.
No, B cell is not get. So here is the thing. Obviously, when you are younger, your cells are eliminating, but B cells as such, they do not get denatured. See, the same if they are healthy people. But if you go to a lot of other diseases in our 80, 90, so if the body has other diseases, the person, irrespective of the age, person equally healthy, there may not be any difference. Is healthy.
There's a condition.
See, the thing is that sometimes our body, like me and someone else my age response, my body can take it without getting a fee more than mine or vice versa.
My concern was whether both of the antibodies.
It need not be the same antibody. Your body may make an against this, and nothing matters. It is the same target. So what site it is binding, it really doesn't matter. As long as it is picture, and you are 20 years old and 20. Suppose there were no memory cells. Suppose, hypothetically, different antibodies, and it doesn't matter one way or the other. It's exactly, and that is it. Start now. Okay. Have you, how many of you have heard about stem cells? For 15 minutes without break, can you teach it? Just know stem cells, but we cannot teach it because we don't know enough about it. Say that again. Huh? They are cells, yes.
They are possibly pluripotent, so they transform themselves to
More stem cells, and then they can differentiate into many, many different kinds of things. That is what is stem cell. That differentiates is cancer will stay stem cells in cancer. Okay? This one. And right now we have not made the whole organ as yet, but the hope is that one day we can make the liver out of in the lab, and we can replace. In one way, they are completely opposite of each other, because in regenerative medicine, right? So in that way, they are very different, but they really work together, and we'll see that. Survival and plasticity to repair tissues. What is plasticity means? Change forms, more plastic, more malleable, right? Very good. Damage caused by cancer. You can reconstruct a new breast and you can replace it. So the whole, sometimes heart is damaged, so you can give heart patch. So this is what it is. Regeneration is controlled proliferation. Differentiate it, right? And restore the architecture. Cancer is uncontrolled proliferation. There is no differentiation in cancer. So they are opposite of each other. So now we come to stem cells. Okay, so what are stem cells? So they can just divide to make more stem cells, which is very important. If they don't divide, then you will just keep losing stem cells as they differentiate. But they can become intestinal cell, any cell, okay, specialized cell. I've not been able to make the brain, but one day maybe we can do patchwork everywhere, but we have not been able to make the whole thing. So Shivani said pluripotent cells. So what exactly are these potent? If it is extremely potent, it can form anything in the world, every single cell of the body, many cell types, but not all cell types. Then there is a lower level that it is. So what it says is that stem form. So some stems. So when the embryo is divided, right, the sperm comes, the egg comes. That's when it can form anything. And that's important. It is found in the first 4 to 8 cells of a developing fetus. Pluripotent cells are embryonic stem cells. They are usually isolated. Everything is possible. Okay, but they're very powerful. Embryos are involved, right? There are a lot of ethical concerns. Cells were available because if there was some kind of fertilization just to create embryos, and then it became bad. Still lower in hierarchy. So during early development, the inner cell mass forms the 3 germ layers, and coming out from mesodermal, they can form different kinds of mesodermal cells. If multipotent cells, kinds of specialized endodermal cells, so they can form. Then comes pluripotent embryonic stem cells. These are blood stem cells found in bone marrow. And then comes unipotent. Here they have, per kid, you are studying 20 different subjects, and then analogy, but it is just becoming very restricted. You are able to form. Yes, go ahead. I was going to go to the good news, converted to totipotent cells.
So it's like the hierarchy, like
I had not shown that. I was just going to show the good news in justice. That is very important point. I'll come to that also. So whenever you make all these things, cancer is always a risk because it has to be very controlled. Really awesome, right? But I just told you that there are a lot of ethical concerns because people were doing in vitro fertilization. So how do you make, if you make these things, so you don't want to keep on taking out of the body, okay? So how do we take normal cells and you change it to stem cells? So the director of the institute was saying there has to be some way. He is extremely forward looking. He can think 30 years, 50 years ahead. What are those factors that you take normal cells like skin cells, which are completely differentiated, right? And because we were doing it at that time, but somebody else did. So that gave, you take skin cells, they're 100% differentiated, right? And there are 4 factors you add, 4 protein lab in different ways. You add those 4 proteins, they are called Yamanaka factors or Yamanaki factors. Skin cells, which are 100% differentiated, right? And skin cells are easy to give, like you can just take a, you are not going a hierarchy. They are as good as embryonic stem cells. So we have not made the 4 cell thing, but we are still good, powerful, okay? So this is just to remind you that I'm stepping. Ectoderm forms a nervous system and skeleton, skin. The one which is these cells, blue, they will form these organs. Okay. So completely new, which we have not learned so far. Embryonic stem cells, which we isolate from teratoma. Do you know what is teratoma? Teratoma is a kind of cancer which is made of different kinds of, it's not prostate cancer or breast cancer or this is a mixture of different kinds of cell cancer. Somebody was saying about the cancer. So that is teratoma. Adult, where the stem cells are there. So if there is some problem in digestive system, this is hematopoietic stem cells which are renewing our blood all the time. Sometimes when people have leukemia, repair the patient's thing. So there are numerous niches. That's naturally occurring stem cells. You actually don't want to go inside the body. Application. iPSC cells. So adult cells such as skin cells, you reprogram this when you do, OCT4, SOX2, CLIP4, CMYC. Now here is the bad thing. When you make iPSC stem cells, if iPSCs comes with the risk of cancer, okay? And the blood and all the things. So our doctors throw away everything, right? So is it collect people are doing when the baby is born, the parents are stored fluid and placenta, because if they need stem cells in future, it's just discarded before because they're very rich in stem cells. Okay. So there is an egg, a sperm comes, the egg is fertilized. Now this is one, and these are pluripotent because this is not 4 cell stage or 8 cell. The endoderm, and germline. So they are pluripotent cells. Research into developing for diseases like Parkinson, diabetes. Okay, Parkinson, they degenerate. Hippocampus degenerates. Okay. Typically from surplus embryos created via in vitro fertilization. Pluripotent, so they can differentiate into multiple cells. To obtain the raising questions about the moral status of many other lives. Okay. Now let's look at restricted potency. So look at this. Remember, I can only differentiate into neurons, astrocytes, oligodendrocytes. Mesocytes ones, there are many more there. So you will be surprised how many naturally occurring stem cells. So they can be in blood vessels, brain, skeletal muscle, peripheral blood, all over the place. And they are hiding in some niches, like small areas where they are located. Okay. So there are many parts where they are hiding. So stem cell niche and microenvironment, they do their job. Okay. So adult stem cells reside throughout the body and support various tissues because of their stem out, and you can use them. Okay. So they are stored here, and then they are not hiding, located somewhere. The whole microenvironment is helping them stay there in that dynamic structure, which includes supporting ECM. ECM is extracellular matrix. So they have to have, okay, right. So so this is So constituents of membrane-bound factors which are helping them survive. Okay, they have neuron nurturing environment for these stem cells to serve. Hematopoietic stem cell niche. These are muscle stem cells. So this is just the environment. That's where they are doing. So don't get into the details of each. These two guys first before we go to the third type. Adult stem, these are not pluripotent cells. These are fourth cell embryo, then it will be totipotent. Okay, differentially precious due to their pluripotency, but okay. So go ahead. They are not very restricted. So for example, hematoid origin and myeloid origin. So they can make B cell, T cell, NK restricted to where they can go, what they can become. These adult stem cells are undifferentiated stem cells in differentiated organs. They are recycling related cell types. So if it is hematopoietic stem cells, they will make all different kinds of hematopoietic, brain, blood, liver, skin, skeletal muscles, adipose tissue, just to name some of them. These are just blastocyst. Here, 4 to 8 cells is totipotent, but you will get 4 to 8 cells, and then multipotent are the ones we are isolating from adult stem cells. I only tell you about 2 of them. Mesenchymal adult stem cells, actually occurring stem cells which are distributed. Marrow not only has hematopoietic stem cells, they also have mesenchymal stem places. So see, all of these different places they are. So they can form nerves, they can form heart tissue. You can see mesenchymals are very. So if somebody has bone surgery, people who have heart surgery and heart is damaged yet, but we can put the patch there. Okay, and they can also this part is controversial, so let's not go there. Value, this is very important. Immunomodulation, anti-inflammatory, useful. So inflammation is very bad. So when there is inflammation will go down, which is a big thing. Okay, so inflammation needs to go down. Inflammation is very higher. Anti-inflammation, they suppress when there is excessive immune response. They suppress under the right conditions, promoting angiogenesis to support healthy tissue. So these are clinically established. They are one. So GVHD, I remember I told you earlier today, body, they recognize the patient's body as foreign and they tell me that. How? Suppression of? When you are giving an organ, even you are doing bone regeneration, you are giving, has to be controlled so that it doesn't cause any damage. But because it is immunomodulation, obviously they can do bone and cartilage repair, right? So investigational, they are not approved as yet. So people who drink too much alcohol, cirrhosis, autoimmune disease, some people you may have heard of that. So I know of a couple of people, very long recovery during COVID. They were hospitalized and basically their life, they cannot do much work, they get tired very easily. So it's a long-term effect that MSCs can fix it. Okay, but immunomodulatory roles of stem cells, they can promote immune tolerance in many different ways. So it is different from cancer. In cancer thing, right? So we have to stop the immune system from reacting for that one. Okay, let's start the next one. All right. It's on 26th slide. I don't know why it stopped. I'm glad I did not even talk about pluripotent cells as yet. So I will come back to mesenchymal stem cells again in the next. So you guys are very familiar with this figure here, right? So hematopoietic stem cells can give rise to here B cells, T cells, and NK cells. So if you compare MSC's function of mesenchymal stem cells is to regenerate, body can actually regenerate and not reject cells. Can you name one cell, which kinds of immune cells, but name one cell, not suppressor, not activator. Don't check. Extremely important, blood cells. Okay, RBC, cartilage, fat, muscles, stromal cells, cord blood. These are also present in bone. So, what is the use of leukemia patients, right? And so, what they do, that usually siblings have bone marrow, right? This is known for at least they don't die, right? But on these two, now let's go to these exciting iPS. So you know about the embryonic stem cells. And we add four factors, OCT4, SOX2, CLIP. They are better than kinds of lineages, but they can go to stem cells. You take somatic cells, think here. Okay, so they are extremely, you take it, you add them, they are ready to go. Both. You take it out, you add these things, it's not going to be that useful. So suppose you have to fix the car. So they are very good. If you compare MSCs, mesenchymal stem cells, isolate hematopoietic stem cells or mesenchymal stem cells, we have isolated from adult cells. Okay, MSCs are multipotent, these are pluripotent. They can form muscle. These are pluripotent. They can form ectoderm, mesoderm, endotherm, anything. Cells. If you're working with mesenchymal stem cells, die. Okay, but pluripotent cells, okay. So unlimited, that's the advantage. But IPS differentiated, the stem cells can form tumor. May form teratomas. What are teratomas? And it is coming from stem cells, so that's how it is. And hypoimmunogenic, they don't turn on, there's no donor match, it can trigger another person because they don't turn on homologous from the same patient of the use for regenerative medicine to fix them. That's when you use MSCs. It is very popular to do drug generation. I will tell you about organoid in the next. Okay, stability. These are very stable. All right, so this is the summary for that. Ethical issues, none for somatic cells. Multipotent, pluripotent, limited self-renal capacity, unlimited. They don't have tumor risk coming from the biogenic that can cause immune rejection, strong immune response. They are widely used. This is still because they are multipotent, they can form all cell types because they are pluripotent. Okay, so I want to just remind you about this one again, autologous versus allogenic. Autologous recipient. Autologous is from a different donor of the same species, so human to human, not human to pig. And xenogenic is from different species, pig heart valve and humans, right? We will not take a human heart and put it in. Availability, you can just keep it there. No need to harvest patient cells, and the disadvantage is that so they are found in umbilical cord, placenta, amniotic fluid, more potent than adult stem cells, and they are not really discarded after birth. So like I said, now patients are saving those cells. Now earlier they were thrown away, and people who were smart, they were saving it to do it, use it for research. Okay, so these cells are generally contained multipotent, fitting into various cell types, and they also have and this is, these tissues were thrown away, so they were anyway thrown away, so they are non-invasive, pose no risk. Proliferation, they can the activity of the cells which are attacking cells, and they lead to activation of Treg. Remember, Tregs are immune suppressive cells. Okay, and the inflammation goes down, and they can differentiate into, and all the cells which attack, they quieten those guys, and they can differentiate into ectoderm. And now that you know all about stem cells, I will tell you exactly what is regenerative medicines. And so what they say is the fundamental pillars, which are called triad of tissue engineering. So they're called R3. What happens is that you develop a new method, right? And you give it a very catchy name like PROTAC. PROTAC is really awesome, but anytime you develop something, you give it a catchy name, hoping that this will catch up and it will become real. So they are giving it this R3 paradigm there. Okay, so you need three things. Now they actually does the biological material. So to actually iPSC cells, you need scaffolds. Suppose they give it a scaffold so the cells can actually hold whatever patch, so then it has to rear, and then you have to also control the immune system, and signal will tell how to differentiate. So even if you put if you don't give it a proper scaffold, they will just spread. The scaffold should disappear without hurting the system. So this should be biodegradable. Cells are the stars of the show. They are undifferentiated cells that can transform into cells need a home. They cannot float freely. So scaffolds are engineered structure to give the whole 3D framework and then tell the cells to grow there proper. So you have to really give them the proper structure, degrade and get removed. And then you have to have all the growth factors. Tells you that if there is a damaged organ, you replace it. You want to regenerate that part of the liver. Okay, so that is to heal itself. Okay, so now the fourth pillar is, so these are the three pillars there. Okay, I did. Okay, I have to learn from you guys. I don't know how to use it. Immune modulation is part of them. So the goal is that these are the diseases. It's definitely doable one day. You can create a whole organ, and instead of getting it to the other patient, so you can take and then you can put it back there. Okay, so stem cell therapy, also acute liver fibrosis. Okay, so this is just different kinds. Now keep in mind, if you make CAR T therapy, it becomes regenerative medicine. If you take out the T cell from the patient and make CAR T, then it is not then it comes under regenerative medicine. Okay, so that, okay, you can make any kind of cell. You can make CAR T, you can make macrophages, you can make CAR M, CAR N. Double check if it is still the last one. Going to be ready up until 1 o'clock. So after a few minutes, we can 3:30 or 4, latest by 4. Okay? Some questions before I go to the next one, the last part of stem cell therapy. The good news is that this is the last day when we have 4 lectures. The last day we'll only have 3 lectures. This is broken, that's why it is fault. We always had 4 lectures except for the first day and the last day. It just happened, right? First day is last day. Okay.
So my question is, usually the vaccines activate immune system. So sarcoma, where HIV infection leads to or any other autoimmune conditions are.
But if it is a immunocompromised patient, you have to be very cautious about giving a vaccine. You don't want to create a little bit of healthy body. Yeah, you have to have some immunity. So I would give PDL, like they tell you, you cannot take 2 vaccines at the same time. One time I had to take 2 vaccines, one shot, and they refused to give it to me. So that tells you that you are giving one immunity, and yeah. So yeah, go.
Both the alleles are deleted. It's like that both parents are having.
Olaparib to inhibit part 1 DNA and then give olaparib. Okay, checking. And it kept saying that when is, but at least it was not showing up in literature. Remove the tumor suppressor. Today I was telling you about synthetic lethality and what protein was that. After TB, MTAP. So there is a protein called CDK, and MTAP just happens to be next to it. So that gets deleted. So what has one deletion of BRCA1, but the tumor gets rid of both. And then you can use synthetic lethality. If the whole body is BRCA1 deficient, again, I did not find any chemotherapy to them. You are just killing that person. So they are not going to get that thing. But oftentimes, like I said, it's one. By any chance, if the cancer has deleted BRCA1, BRCA2, then it is good news. Rest of the cells are happy. Go ahead. So what you do, you take skin cells and you take the vector and the proteins in a cell line, viral vectors.
This MTA, thing, I was confused yesterday also. Confused
Oh,
yesterday
this is a very
also.
confusing thing. I had to read it
Yeah.
5 times before it became clear to me. So
Yeah.
that is a very
Coming to the point, it is synthetic lethality. We give
no, it is not that. It is not that. So what is if you hit another gene in the presence of the first gene deletion, then it becomes that all the so in humans in a cancer where most of the body is expressing MTAP except the triple problem there. So all the cells in the body are expressing MTAP. What MTAP that is catalyzed by MTAP. The methionine is then converted to SAM. SAM is a methyl donor. Okay, so many different. One is tetrahydrofolate and one is SAM. Okay, so it uses SAM to methylate arginine and it is involved in many different pathways. We were working with Pierre. He published many papers on PRMT5. So we were very good friends. So we were cancer. So I had invited a very famous person from Harvard and she was working on all this him and I put him in my schedule so that he can meet. So I called him you later. So he was in an oncologist office at that time, which he didn't tell me. So he was very healthy, food, really, and he was in his 50s. Okay, so years ago, something around that time. Yes, 4 years ago. So what happened is that he said, I will tell you later. And he's the one I was telling you, immunotherapy was not approved at just all the paperwork. It was too late. And he did not live past 6 months person. So we were working on PRMT5 together with him. And after he died, working together with him. And then we stopped working on it. But the bottom line is that app is deleted. MTA levels increase because MTA is not converted to methionine anymore. Makes sense? 20%. So 10% activity is left. So cells cannot deal with it and they will die.
Okay. MTA is
They will inhibit PRMT5. So normal you guys have used inhibitor, it may not inhibit the target, but if you use a lot of inhibitor, it will depends on IC50. But if it is way inhibit LD's toxicity, lethal, that caution, it may be non-specific and hitting other targets also high. It inhibits PRMT5. Now there is barely enough PRMT5 active. Makes sense? That is what is that there. Why don't we fresh to take new information in? Okay, so any other questions? Yes.
You told कि like there will be a signal. So if they got a signal, that is there like some cytosolic determinants that is different.
Is that you take stem cells? You take the skin. Suppose you, I, I have read if you put it at the right location, all those do it in the lab. We know if you want to make go along, right? So if you give the right signal, go into neurons. So those things are pretty much obviously they are missing information there, but depending on the signal.
Is there any some cytosolic determinants like the hemicellular compound, topicotin cell? Then they will like lots of cytosolic determinants in topicotin cell.
Never thought in that direction, but I can tell you. So you, if you at that time, there are 2 cells only. So often they are becoming 4 cells and 8 cells. So initially, it is all cell is secreting some factor which is regulating the other factor, and come back all the way to human body, right? So we the rest of the body, something is secreted from the pineal gland, which is glucagon and insulin and controlling. But to answer, going from the real zygote, yeah, secretory cells to release insulin. Some cells will different kinds of cells to secrete different kind of things. EGF is secreted by some cells and EGFR is expressed. So yes, yeah, ultimately at the very beginning it is coming from the
determinants which are present in the cell, they initiate these signals and they lead
way the embryo develops. It's very complex process. So we don't understand everything, every step, all the things which are secreted and the higher. We cannot do that. We cannot even make an organ ourselves, right? So we have huge amount of missing gaps there. For example, if you have so suppose we have to make immune cells. Okay, we know some things here and there. We can make insufficient thing. We cannot regenerate the whole 3D structure. We have been able to do simple things so far. iPSC cells are extremely new things. Very keep them alive. We are just trying to differentiate them. So people are doing this pretty routinely, but casually here and there, but we are not very good in that. In Japan, they have been able to make. There is one other thing I want to tell you. So keep in mind, okay, so what every cell in the body, forget about the embryo, in even ours, this thing, such different things, cells have something common. So those proteins are expressed, the glycolysis proteins are ubiquitously expressed because every cell needs energy, right? How their specific proteins are expressed in specific cells of the body. How do question comes, egg came first or the chicken came first, right? At the end of the day, the whole magic is within the egg and the sperm. The job of doing this and the others are taking the job of doing that. So I should not disregard RNA because RNAs are the ones. We can go now, all right. So let's go, and then so 150 by 3 will be, we will be done, and then you can package oncolytic virus within mesenchymal stem cells and they will go to the site. So mesenchymal stem cells are very powerful as delivery vehicles. They naturally home to tumors and inflamed tissues, making them ideal Trojan horse carriers for anti-cancer agents. Do you guys know what is Trojan horse? Can you tell me? No, where did this whole thing originated, Trojan horse? Do you know the history to know where the Trojan horse was? Can you tell me? The Greeks were involved in a war, and I don't know with whom, huh? They had a wooden horse filled with soldiers, and they left it outside the enemy's boundary line, and then they opened the door and they saw a nice wooden horse, so they took it inside, and inside were the soldiers, so they just came out and killed them, right? So that's called Trojan horse. So anytime you have to deliver a drug to the tumor site, you can pack it in a Trojan horse and then it will get inside, right? So that's where mesenchymal stem cells are very important. You can deliver cytokines, you can deliver products, converting enzyme, oncolytic virus, nanoparticles, anti-angiogenic factors. So this is very good. So why do they go to the tumor site? Tumor cells secrete signals like CXCL12 and VEGF to recruit blood vessels. So MSCs have receptors for like CXCR4, which binds these ligands, and so they go to the tumor site and they home there. So now we can load mesenchymal stem cells with a lot of different kinds of things, and they will take it to the tumor site. So it's a delivery thing, okay? So suicide gene therapy. So we talked about suicide gene therapy. Remember, we gave 5-fluorouracil, and then we give the cytosine deaminase gene. So you can put the DNA for cytosine deaminase gene. It will go to the tumor site and it will get expressed in tumor cells. And if you give 5-fluorouracil, it can go in the blood, not harm normal cells, but the tumor cells will convert 5-fluorouracil, you give 5-fluorocytosine, not uracil, and when you give 5-fluorocytosine, the amino group of cytosine will get deaminated by cytosine deaminase, and 5-fluorouracil will form. The structure of uracil and thymine are very similar. So it will be taken up by DNA machinery to incorporate instead of thymine, and that is toxic for the DNA. DNA damage will occur and tumor cells will die. Okay, so that is, so this is high local chemo concentration. This is a chemo drug and low systemic toxicity. You can, so again, we are very fond of oncolytic virus. So you can deliver oncolytic viruses. So oncolytic viruses are often cleared by the patient's immune system before even they can reach the tumor. So you can hide the virus inside mesenchymal stem cells, and it will protect it from antibodies during transport. Once inside the tumor, the stem cells burst, releasing the virus, and it will kill the tumor. Okay, so oncolytic virus can be packaged in mesenchymal stem cells. Now let's look at hematopoietic stem cell therapy. So these are very useful for leukemia patients. Okay, so bone marrow regeneration after high dose chemotherapy radiation. So you can even take allogenic hematopoietic stem cells for leukemias, lymphomas, multiple myeloma patients, and then you can inject it in these patients. And as I said, usually they are taken from siblings which have similar HLA. So you have to do the matching here. And once you do the matching here, you don't have a choice of taking it from the patient and putting in the patient because they're all destroyed. So you take up from a sibling and then you give it to the patient. Okay, and that helps in bone marrow regeneration. Okay, then the next is immune engineered stem cells. So immune engineered stem cells, whenever the word engineered comes, that means you are changing the immune cells. Can you tell me some ways of changing the immune cells? CAR T therapy, CAR NK therapy, CAR M therapy. So if you take the T cells out of the patient and you engineer it, that is not stem cell. But if you take iPSCs and you make them in T cells and you put the CAR there, that is stem cell therapy. So iPSC derived NK cells, and then you make the CAR there. iPSC derived T cells, and then you put the CAR there. You can even make macrophages. Okay, so there are different ways. So you are taking healthy donors, and you can treat. So this is stem cell therapy here. So you are collecting the blood, you collect the T cells, you make cytotoxic T cells, you reprogram them. So this is conventional T cell therapy, but here you are taking iPSC cells. Who was asking me about this during lunch? Somebody was asking, you were asking me. So you were asking me, how do you differentiate? No, it's not the antibody thing. So before I come to the antibody, iPSC cells, they are stem cells. They can make any cells. So like you said, we have pretty good idea of what kind of factors we should use if we want to differentiate to skin cells or pancreatic cells or beta islet cells which produce insulin. So if you know all the factors, you can add those factors and you can differentiate them in those cells. Okay, and then you put back in the patient. So we can add similar signals to make T cells, B cells. If we make B cells, we can make antibodies, right? So you regenerate T cells and then you can put it back in the body. So these are CAR T cells. We already know much about CAR T cells. So if they are coming from stem cells, then it is a regenerative therapy. You are doing it. If you are isolating it from the patient or healthy thing, then it is not stem cell therapy. So taking patient's T cell and genetically reprogramming is CAR. But if you take it out, you take iPSC cells and make T cells, then it is stem cell therapy. Okay, so you can make natural and you can make NK cells also, CAR T cell, and you can also engineer stem cells. So you can make stem cells do different kinds of things. So you can take an antigen, okay? You take stem cells and you make natural killer cells out of it. And these natural killer cells, you train them to fight against certain antigen. And then you put it in the patient. So NK cells will go and find the tumor and kill it. Okay, so that is completely stem cell because you are actually making NK cells and then you are training the NK cell. So it's like you are getting a child and you train it to fight against a certain thing, and then you send the person to the war. Okay, so those are immune engineered stem cells. Tissue engineering, as the name suggests, like somebody, the breast has been removed, now you can completely regenerate the breast. That is a very common thing to do these days. So what you do, you take stem cells from fat or bone marrow, then you give the scaffold, then you put the scaffold, then you give enough room for the blood vessels to go, then you give the right signals so that they differentiate into different kinds of breast cells, and then they form the whole thing. Okay, this is just regular CAR T cells. So you can do tissue engineering. So the trick is that you have to give a right scaffold, the right shape. Okay, and then you give the cells, and then you give all the signals, and then you have to make sure that the blood vessels are also doing it, because it has to feed the cells. So it is a pretty complicated process, but people are doing it all the time. So you have to use cells, biomaterial scaffolds, bioactive molecules, like you were saying. These are the signals which will allow the cells to differentiate into breast tissues. Okay. You can use iPSC derived T cells. So CAR T cells is very expensive, over 400K. iPSC T cells, I actually checked the price of this because yesterday we were discussing the price. So it is estimated it would be about 100K. So 100K is still a lot cheaper than taking the T cells out of that. Okay. So iPSC cells have huge potential for cancer treatment. Imagine you can use iPSC cells from the patient and make CAR T out of that. You can make NK out of that. You can make CAR M out of that. You can make NK cells which are not CAR but trained to fight antigens. Okay, so there is a huge, huge potential here. Okay, so iPSCs can be used for cancer treatment. So here there are few examples. So PBMCs are blood cells, peripheral monocyte blood cells, and they can differentiate into many different kinds of blood cells. So you take out those, you can specifically isolate whichever cells you want, T cells, B cells, then you enrich it, you express CAR, you give it to the same patient. So this is the same patient, no rejection. Or you can do allogenic. So you take it from healthy donor, you make different kinds of, you can isolate different kinds of cells, you can make different kinds of CARs, and then you can now give it to multiple patients. So what will you do if you are doing allogenic? How will you make sure that it is not rejected by the patient? What receptor? HLAs. You will remove the HLAs, and you will keep some HLA which is recognized by NK cells as self, and then you will also delete TCR, right? So then you can give it to patients. Here we are taking immune cells or skin cells, other somatic cells, we make iPSC, and we directly differentiate it, and you can make simple NK cells. T cells are CAR, but these are NK cells. NK cells are killer cells. You can make it and you can give it, okay? And that will really help. Now, as I was telling you, in Japan, iPSC cells have been used to make retina. It is approved in Japan only so far, okay? So this is pluripotent cells. These are obviously embryonic pluripotent cells. This is fetal derived cells, and they have been reprogrammed to make stem cells, retinal stem cells, or brain or spinal cord. Okay, and you can make different kinds of tissues out of it. So in Japan, they replace retina of a patient using stem cells. Okay. So it has not been used for diabetes, but research is going on to, the islet cells are the ones which release insulin. So the idea is to make use pluripotent cells to make these beta cells which increase insulin production. So this is a potential use because it has not been realized as yet. Most of the things have not been realized as yet. Things are going on. Now, stem cells as tools to study cancer. So organoids, avatars, and chips. Okay, so let's look at it. What is it? So do you guys know what is an organoid? So when you guys are working in the lab, and suppose you're working on cancer, you are using tissue culture dish, right? So it's two-dimensional. You are plating the cells and they are growing, right? But the tumor is three-dimensional. So these days, if you just do cell culture like that, it is not acceptable. You have to use the three-dimensional structure. So that is called organoid. So Venkatesh lab developed this very easy way to make three-dimensional spheroids kind of thing, which are very useful just by using the simple comb in gel. It is just fantastic way of doing, and they have very beautiful confocal microscopy. If you treat it with drug, it's just really beautiful work. So one student from his lab came to my lab and she created spheroids in my lab also. So they are called organoids. Okay, so let's look at them. So usually we use 2D cell culture, so that does not mimic 3D structure. So when you make an organoid, right, then there's the oxygen gradient because inside will not get as much oxygen as outside. But when it is 2D, everything is getting enough oxygen. So if 2D does not really mimic the tumor, which is the 3D structure, if you do mouse model, it's very expensive, okay? And then mouse immune system is not the same as human immune system. So look at this. This is a 3D structure here, okay? The one your student made looked a lot more beautiful. This is very ugly. The way you should look at is his data, like his spheroids are extremely beautiful spheroids. Okay, so these are called organoids. These 3D structures are called organoids. So they are used for many, many different kinds of studies. And nowadays, what you can do, you can obviously make organoid after the cells. But you can, the more important thing is that you can take out small tissue from the patient, which is available because, you know, patients undergo surgery all the time, biopsy all the time. So these patient tissues are available, and you can make organoid out of the actual tumor cells from a patient. These are called PDX models, patient-derived models. And if you do drug screening in those compared to cells, that is more effective. If you do cancer studies in those, that's much more effective than just using 2D gels, 2D cells. Okay, so patient-derived organoids for drug response. Okay, so comparing 2D with 3D, so these are patient-derived organoids, that's PDOs. So these are flat monolayer on plastic. These are complex 3D structure like a mini tumor. And here, because they are flat, they have huge access of oxygen. They don't have to compete because they're happy flat things. Here, depending on this thing, these guys will have more oxygen inside. They will not have enough oxygen. Okay, so and if when you grow 2D gels, they change over time. If you have too many passages, they change over time. Usually 3D structures are more stable, the organoid structures. So they have high correlation with actual patient response. Something we do in cells will not work for the patient, but if you make a 3D structure, it will work for the patient. So you can do a lot of things. Organoids can be used to study as a disease model. Like we work on prostate cancer, so prostate cancer organoid will really mimic prostate cancer, right? You can use for drug discovery. If you, like say 96 well plate and in each you have organoid, if you can do a drug response there and see how effective it is. Okay, it is used for personalized medicine. You actually have the patient sample, you can look exactly what is going wrong there. And it can also be used for regenerative medicine. Okay, you can expand them. You can even, you can do a lot of stuff from organoids. Okay, so this is tissue engineering, regenerative medicine, cancer research, all of these things, drug screening. Okay, and then you can isolate the organoid. You can break open the organoid, and you can actually change the cells and then again create the organoid. Okay, so there is a lot of things there. So patient-derived cancer organoids, you do a biopsy, and once you have it, you can again create a tumor organoid. You separate the cells, you replate them, and you can create that. And then you can do quite a few things. You can take normal cells, you can edit the gene, and you can create a tumor organoid. Suppose you delete p53, it will become tumorigenesis. Tell me other ways you can take normal cells and make it tumorigenic. Any method. You overexpress aurora kinase, it will become EGFR overexpress, it will become tumorigenic. Anything else? Absolutely. Overexpress make becomes oncogenic. These are the questions you're going to get. Okay. All right. So applications of patient-derived organoids, personalized and precision medicine. Just imagine you are actually taking the tumor out of that, you're creating the 3D thing, and then you are actually doing it. So one of the things, obviously, you can do DNA sequencing, you can look at RNA, you can look at proteomics, find what's wrong. But you can also do the drug response. You give the drug and see which drug is working in the 3D structure. That is something we have not covered so far. So that is drug response prediction is very accurate in 3D models. Okay, and suppose you take out the biopsy from someone and this tumor is very heterogeneous. Now in your organoid, the whole heterogeneous population is there. So you can see what combination of drug is working, right? Okay, so it is very important in studying tumor biology and progression. It will tell you what leads to tumor initiation. Because it is 3D structure, you can even mimic the immunosuppressive environment there. Okay, and you can test your therapy that way in the immunosuppressive environment. Okay, so advanced organized systems allow co-culture with other key components of the TME, such as fibroblast, endothelial cells, immune cells. And you can create a real structure in a dish, and you can study it that way. So just imagine how powerful is this system as compared to just using pure cells, pure one cell line which is immortalized, right? These cells are not immortalized, these are actual patient samples. So they're very effective. Okay, you can actually do metastasis research. So that was organoids. Let's look at avatars. Avatar is just a word they have stolen from us. Okay, you know avatar, right? Avatar. Okay, so avatar is that you are basically creating the whole tumor in the lab, right? In the 3D form, and then you are doing drug discovery with that. That's what is meant by. So that is you test 50 different chemotherapy drug combos and you see which one works. Okay, so instead of probability, it tells you deterministic. Okay, it is much more accurate. Then I have a friend, I was trying to remember his name, Dan something. So he studies metastasis on a chip. So without going into the details, they have this kind of chip they have made, and there is something flowing there. And that is very easy. That is a very nice way, quantitative way to measure metastasis of cells, how metastatic those cells will be. Okay, so then you can even check the drugs to stop metastasis. Okay, so small chips with microchannels light with living human cells, and it stimulates blood for tumor cell interaction, drug delivery flow. So you can create a whole cancer on a chip. Okay, and you can also use it for measuring metastasis. Don't go in the details there. So metastasis causes 90% of cancer deaths, yet it is hard to study in 2D. So chips can visualize cancer cells crawling into blood vessels. Makes sense? And watching cells exit vessels in very distant tissue chambers on the chip, you can see everything in live photography. Okay, study how micrometastasis sleeps for years before waking up. So what happens is that sometimes tumors go and lodges at different parts of the body, like real micrometastasis, and then they will be quiescent for a while, and then suddenly they will become active. Okay. All right. The third part, stem cells for drug discovery and screening. I have already told you about that. So like you were saying, right, tubulin inhibitors. So you take iPSC cells, you differentiate them into cancer stem cells, and then you do the screening and see how it works. And you can make it very aggressive cancer stem cells or epithelial mesenchymal stem cells, and then you will know how effective is your drug. So it can be used for high throughput organoids are used for high throughput screening. What is the meaning of high throughput screening? Can you tell me? Anyone? What is high throughput screening? We just had lunch, so you guys are tired. We all need tea, which we will get at 3. But what is high throughput screening? Yeah, that's what it is. It is high throughput, right? So how many compounds make a high throughput library? You guys are in 2025, right? What is a high throughput screening? Like how many compounds? 100, 200?
Now it's a very huge chemical space, so you have to apply various filters to reduce the chemical space.
No. How many compounds? Just keep it very simple. How many compounds will constitute a high throughput screening? Here is the answer. It can be 100 compounds, it can be 100,000 compounds, right? So we used to do it in 96 well plate, which all of you have used, probably have seen at least, right? So in 1999, and this is 1999, 26 years ago, right? I joined Novartis, and the director of that place hired a person from one of the biggest car companies, Ford or something, and he was an engineer. So he said, make a high throughput machine. So he built a high throughput machine at Novartis, and that was the first one of its own kind in the entire world. And in 99, 2000, we were screening 2 million compounds in less than 1 week. And you guys, we talk about 96 well. He had, in the same area as a 96 well pit, we had more than 3,000 wells. And there were nanoliters of compounds and cells, and we would sit in our office and the data would come to our computer. And then Peter shows, he had many spin-off companies with these high throughput facilities. So nowadays you can imagine what high throughput screening means. So you can make organoids and you can screen billions of drugs that way. Okay, you can identify novel targets and resistance mechanism. So one of the ways, let's say you develop a new drug, right? And it is very effective. And somebody asked me, what if it develops resistance, right? So what will you do to be ready for that resistance? So what you can do, suppose your drug works very well, Vanshika. So you can take your cells or organoids, and suppose it works at 1 micromolar concentration, right? So you give 1 nanomolar concentration or 10 nanomolar concentration. So some cells will die, but some will survive. You keep them in that drug for a very long time, like 2 weeks. Slowly, whoever are surviving. Then you change from 10 to 50 nanomolar. Some cells will die, but other cells will become resistant, and then they will divide. Then you increase it to 100, and then you give them some time. So this way you are slowly increasing the drug concentration, and 3 months, 4 months, at 1 micromolar, those cells will be resistant. Then you look what is different in that cell line. That will give you the preemptive, in advance you will know what kind of drug resistance your drug is likely to develop. And if you use it in organoid, it's going to be better than flat cells. So you see that you can predict cancer resistance mechanisms before it happens in patients. Okay, you can do toxicity, whether this drug is going to be toxic or not toxic. Okay, then stem cells in regenerative support during cancer therapy. So this is when you give cancer therapy, it is pretty toxic. It can lead to cosmic deformity, functional loss, like speech, movement. People who have neck cancer, their voice changes after surgery. Okay, so they can actually affect the speech. Radiation leads to a lot of fibrosis. hardened damaged tissues. So regenerative medicine can fix all of these things. Obviously, breast replacement is one of the deformity thing, right? So core biological functions. So you can build biomaterial scaffolds for tissue repair. So first thing is that if you have to build an organ, you have to provide a support. That support should be non-toxic to human body. We don't want to put anything which is toxic. The person will die. Plus, you provide the support, but once the cells, so it has to be biocompatible, and once the cells grow and everything is there, you want the support to disappear. Correct? You don't want that artificial support to be there. So they should be biodegradable because then it should be all pure tissue based thing. So that's what it is saying. You have to give a temporary ECM like framework. ECM is extracellular matrix like framework. To maintain the slope, you have to give the right biochemical signaling, and then you have to put the stem cells to survive, proliferate, and differentiate into the tissue, the kind of tissue you need. Okay, it should be biocompatible, it should degrade after the whole structure is formed, and then it should have the same mechanical properties. So if this is stiff, it has to be stiff. If this is soft, like bone is porous, so it has to really mimic the bone structure. It cannot like a it cannot be an iron rod there, right? It has to have the porosity so that it can get the nutrients, the blood vessel and everything. So it's a challenging thing. Okay, so this is just telling you, you don't have to remember anything here. Like kind of polymers that are used, collagen is present in us, right? So they use collagen, all the things which are present here, synthetic polymers, decellularize. So they take the whole ECM, but the cells are there. So they get rid of the cells, but they have the ECM. So they use that as a support because that is very natural support. Okay. Okay, and then they control the pH, they control the enzymes, everything there, temperature. So this is just breast reconstruction. This is very popular, but it is super, super, super expensive. Okay, this has been done in patient. So if there is a kind of stroke or some kind of damage in the heart, they are able to patch it up. They cannot build a whole new heart, but they can patch it up. So this is called cardiomyocyte patches. So they have patched it up. Okay, they have been successful in regenerating the bone. So that's quite a bit of success. You seed MSCs to stimulate bone formation. You get BMP2 to release accelerate healing. Okay, head and neck, resecting tumors in the jaw, tongue affects breathing, eating, and speaking. So they have regenerated these kind of things. It's quite a bit of things after cancer therapy. No, trachea, this thing, yeah. So they can do that. So high risk experimental. So this is experimental but life-saving for non-resectable cases. Okay, so that was that. Let's quickly go through these two things now. Gene therapy. You guys know all about gene therapy, right? You put the gene inside and you fix the thing. So here it's a really beautiful thing which has been actually done. Okay, so if there is something wrong, you can use CRISPR-Cas9 and you can fix it. If you had a choice of doing gene therapy, can you suggest some disease where we can do gene therapy? Like some kind of sickle cell anemia. It's just amazing. I know you guys don't have this presentation. If you had, the next slide is sickle cell anemia. So this is really, 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 really amazing. Okay, so gene therapy is that a gene is wrong and you fix it and then you give it. But because it is stem cell, you are fixing it in iPS cells. Okay, so this is sickle cell anemia. Anemia. So what goes wrong? Hemoglobin has just one single mutation. So this is normal red blood cell, donut-like, and this is sickle cell. And these sickle cells, they aggregate like this, and they restrict the blood flow. So normal red blood cells, they have a half-life of 120 days, and sickle cells die half 10 days, in 10 days, and it's very bad. Okay, so this has been done. Actually, I checked, this has been done. So patient stem cells in their bone marrow make diseased hemoglobin that make red blood cells sickle shaped. So you take the gene, you fix the thing. So you take the stem cells, you can extract it from hematopoietic stem cells, easy to get, or you can take iPSC cells, you genetically modify, you give it back to the patient, and now it is normal. And it actually works. This is not experimental, this actually works. Okay, so this is just to show you that it has huge potential. You can take skin cells, you can correct them, and what is the beauty? Every cell has the same DNA. So you can correct the DNA, differentiate, and give it to the patient. Okay, this is hematopoietic stem cells. So this is hypothetical, but you can engineer CAR T NK cells, you can give it to the patient, you can take stem cells and make any kind of cell, and you can edit it, right? You might have seen some science fiction shows where they're making superhumans, like really correcting everything. Okay, all right. So if you want to turn off the immune system, you take mesenchymal stem cells, and that take care of the immune system quietening. So if you have to regenerate something or give new therapy where you don't want immune system to be super active in those cases, so then you use mesenchymal stem cells if you have to reduce the immune response. Okay, you can take mesenchymal cells and you can make so many different kinds of cells. Okay, the last thing, what you are interested in, cancer stem cell therapy. So these are not good guys, these are bad guys. They are not originating from iPSC cells also. So cancer stem cells, so remember I keep saying that you take a tumor and you irradiate it, and 99.99999% cells will die, but 1 or 2 cells will alive. And they are extremely resilient, and they are usually cancer stem cells. So that one single cell is so resilient, you cannot kill it. And that cancer stem cell can form a whole new tumor in all different parts of the body. Okay, so that is that. Here they are showing you many cancer stem cells. So these are cancer stem cells. The rest of the tumor died. These are the persisters. Okay, and then they can form really extremely aggressive cancer. So you can, so why chemo fails? Because CSCs, they are often quiescent, so they will become very quiet, they will not divide. And if they're not dividing, chemo will not work, because chemo depends on cell division, right? It is changing. So they will become quiet, they will exit cell cycle, they will come to G0 state, like they are sleeping. So chemo drug is not going to work on that, because it depends. They are mitosis inhibited. If there is no mitosis, nothing will happen, right? So they will become pretty quiet. And then there is tumor heterogeneity. You can kill some cells, you cannot kill other cells, because cells are different. Tumor growth kinetics, if somebody was saying that cold tumor versus hot tumors, if they are growing slowly, chemotherapy will not work, because rapidly dividing cells die faster. Okay, when you have MYC or P50, MYC overexpression, P53 mutation, that will also be a problem, because whenever tumor suppressors are gone, oncogenes are overexpressed, they can become resistant. So there are many different things which can give rise to cancer stem cells. CSCs are cancer stem cells. So how will you target cancer stem cells? So there are many ways to target it. We know what proteins are upregulated, and you can give inhibitors of that. But because we are studying stem cell, a very nice way is that you change stem cells to differentiated cells, so that they don't come back. Once they are differentiated, they don't come back. So if you differentiate it, you can use ATRA, all-trans retinoic acid, and they are differentiated, then they cannot divide and make more stem cells. They are gone. So you basically do that. Okay, this is something I added for you guys. So remember, we keep talking about autologous versus allogenic kind of things. So this is for you guys, that stem cell therapy is very successful already. So I said, okay, show me the successful therapies. because most of the things are still investigational. So tell me the successful thing. So these are the things for which stem cell therapy is actually very effective. So for ALL, acute lymphoblastic leukemia, multiple myeloma, CML, relapse lymphoma, these all are treatable by stem cell therapy. So remember I was telling you about an undergraduate who was in my lab who had leukemia? So I never asked her what kind of therapy she got. So she was undergoing therapy every 3 months, right? And I told you she wore a lot of makeup to cover it up. And then she went back to Singapore and she had some therapy and she said she's cured for life. And I really wanted to ask her, but I never asked any personal questions because I was the PI. If I asked her, she would have replied, even if she didn't want to share her things. So I didn't ask her. But now I'm thinking that she got stem cell therapy, and that's why she was cured. Because all these years I've wondered what therapy she got. So this is very successful for all these blood cancer, disease-free survival, complete remission, long-term remission. It's just really, really good. So it actually cures it. So stem cell therapy can be really phenomenal. That's it. We are done. Not bad. बंद कर दीजिए. अभी तो चाय तैयार नहीं हुआ होगा ना. 3 o'clock. So ऐसा करते हैं ना कि हम लोग अभी ट्यूटोरियल कर लेते हैं 20 मिनट 22 मिनट, फिर हम लोग फिर 3:15 1 hour. So 4:15 हो जाएगा. हम लोग का 15 मिनट वहाँ सेव हो जाएगा. ठीक है. फिर आप लोग थोड़ा 15 मिनट पहले रिलैक्स कर सकते हैं. तो मेरा फिर कार को 4:15 कर दीजिएगा. थैंक यू वेंकटेश. ओके. हाँ ठीक है. क्वेश्चन लगा दीजिए. हाँ आप लोग क्वेश्चन पूछिए. Did you guys follow everything? Did you follow everything? Okay. So the good news is that stem cell therapy is very useful. It is very, very useful for people who are getting leukemia. But I have a friend who is a doctor who works on leukemia, but he's also a scientist. And I recall, I wasn't very interested in leukemia, to be honest, at that time. But I recall he says that two things. बच्चे लोग, the children, they get leukemia much easily. Most of his patients are, children. But he also said that they get well soon. So that's good news. I have seen that leukemia used to be a death sentence. If somebody had leukemia, the person would not survive. Exactly.
Ma'am, actually, that availability of payments is also improving nowadays. Because in India,
You should. It's getting charged here. So in India, the donor availability is increasing also now.
I'm not able to remember the name of the one. PhD कर रही हूँ.
अब आवाज नहीं है.
One of my colleague, he has recently submitted one swab sample for it is one lab DKMS, I think some lab in Dresden, Germany. So they have their labs in India as well. So when we submit our swab sample, they identified I guess they have some HLA testing or something, and
they store it in a database, and when there is any patient and their requirement, and if our sample match with it, then they call us, and even they pay for our traveling and everything if we want to, if we agree to donate our stem cells, especially in case of leukemia. Yeah.
Yeah, stem cell donation has been there for a long time, like very, very, very long time. No, so what they do, they take out the bone marrow, and stem cells will go along with those. They don't separate the stem cells. So earlier it used to be called bone marrow transplantation only. So they're taking out the bone marrow. So how much bone marrow do they take out? I know it is a very painful process, but the thing is that if you can save someone's life, it is extremely good. It is extremely good. If like the person who is donating, even if the person suffers something, but it is at the cost of someone's life, it is worth it.
Ma'am, but I have one question. Like in case of hematopoietic cell, we can directly take it from blood sample and process is
Not blood, bone marrow.
actually they have one video on their website where they are showing us the procedure. So there they are simply taking. There is one machinery, and they're simply taking our blood, separating the plasma and stem cell and normal blood, and transmitting back, like giving the patient its blood after removing the cells.
No, so here is the thing. You definitely can take the blood samples, blood, and you can isolate the T cell. Actually, you are going to the lymphatic system and blood also possibly. So they call it leukophoresis or something. So I'm thinking it is the lymphatic system, but it's almost the same. But they are isolating T cells and B cells which reside there, right? So that could be for engineering CAR-T or just weaponizing them and sending it back. But if you have to take the stem cells, I'm not an expert in stem cells, but I think you have to go to bone marrow. I mean, there are so many niches, like I said, but bone marrow is very rich in stem cells, very, very rich. So, bone marrow transplantation is different from blood samples and isolating T cells. But keep in mind, if your HLA is matching with someone, and so you are a healthy donor of T cells, that's fantastic. That itself is very good. But if they are taking stem cells, most likely they will use bone marrow cells. So when there is leukemia, right, you don't take blood and you just transfuse blood. You also do bone marrow transplantation. So many people, I mean, I don't know personally, but I've read about many people that they undergo bone marrow transplantation because it is the bone marrow which is making all these immune cells and the red blood cells. So they completely die because of the radiation therapy or chemotherapy. So whenever you give chemotherapy, even if it is not blood cancer, if it is any cancer far away from blood, bone marrow is the one which is most affected because they are very rapidly dividing cells. So any chemotherapy, bone marrow is affected. So especially for leukemia, it's just really bad. So yeah, there are so many things. So they could be isolating blood for something else also. But I have just heard that taking out the bone marrow is painful, but I'm sure the person is unconscious.
Ma'am, then in case of isolating mesenchymal cell, there also procedure will be difficult.
Mesenchymal cells are also produced in bone marrow, right? Mesenchymal cells are also produced in bone marrow and other places. So yeah, whichever way they take out, I am assuming, I'm not a doctor and I don't know how these things work. But the thing is that it seems bone marrow provides a lot of stem cells. So you can directly go there and get it. But iPSC cells would be a very good options. Professor Venkatesh was asking me that how about we get iPSC cells. So right now iPSC cells, if you buy it from commercial vendors, it is pretty expensive. Like these many cells are $500. So it is very limited. Like it is super expensive right now. But having said that, people are doing research, right? And once you figure it out, see, everything is expensive when the whole thing is developed, and then people always find cheaper alternatives, right? So it's going to be okay. I mean, first we need the therapy before we think about the cost. Cost will come later. Once you know the way, then the generic things can be developed. The cost comes not because the experiment is expensive. Cost comes from all the failed experiments. You do 1,000 failed experiment to get one right thing. That's where the cost adds up. But when you know the right thing, then it's not expensive to do the right thing. Correct? Yeah. So, but this is very interesting to know about it.
Hello. What is the shelf life of CAR T cells and CAR NK cells?
I don't know. I don't know. I think so. Right now, they are barely at the stage where they are storing it in the shelf. They are making it, they are using it, they are making it, and they're using it. So once we actually make it on large production, then we will know the shelf life. But I'm guessing once you put it in the freezer at -20 or -80, it will be good for years.
Just blood banks
I
like
am sure
we
it
can
will
make
come to
some
that. We'll figure out a
CAR
way
T
to keep them
banks.
happy. We'll figure out a way to keep them happy. See, antibodies are very unstable too, right? But immunotherapy is everyday's thing now, right? So, uh, so CAR T will become a simple thing too. You know, we are focused on cancer completely here, but there are a lot of bad diseases which need stem cell therapy, like macular degeneration in the eye, right? That needs stem cell therapy. Retinal degeneration needs stem cell therapy. Then you know about this disease called muscular dystrophy. You guys have heard of it, right? That's a devastating disease. Like the person is just lying on bed, young people, and they cannot get up. So stem cell therapy can really be useful there. They can do the gene therapy. They have done the gene therapy. So there are four kinds of muscular dystrophy, and there is a kind of muscular dystrophy which happens in boys only, not girls. There is a boy specific thing, and younger children, certain age like limit. So they have done gene therapy on one of those guys, and the boys, let's say it is 8 years age. The boy was turning 9 like 4 days after. So the parents really pushed for it before the birthday. It's very crazy. The rule is 8 years, it cannot touch 9 years. So they did the surgery like 2 days before his birthday, and he was fine. He was the investigational case. So, but it is not done for many other groups of muscular dystrophy. So there are so many diseases. No, for that particular, I don't know why that treatment they say that it should be less than 8 years. Yeah, or 9 years, something. If you check the muscular dystrophy gene therapy, the whole story will show up. But there are four kinds, to my knowledge, four kinds of muscular dystrophy. So the most prevalent one has not been uh, treated as yet.
So, ma'am, my question to you is when we were discussing about mesenchymal stem cells, so they do immunomodulation. That is, they shut down immune system for a while. Does that means our body holds a gap between creating something new before it comes under full power? Because is it so? It is so. We can exploit it.
So I'm not following the whole thing. Can you repeat it again?
MSCs do immunomodulation.
Right.
That is, they shut down immune system for a while.
That's right.
Does that means our body holds a gap between creating something new before it comes under full power? Because if it's so, we can exploit it.
No, so here is the thing, if the body is doing by itself, right, it does not need to shut down the immune system at all. It is only that when we are giving something from outside, we have to, immune system is not supposed to be shut down at any time. No, no, no. So we are the one who are forcing immune system to shut down. So suppose somebody is given kidney transplant or whatever transplant, right? Like people donate their kidneys and all these different organs, right? So if you do that, then you have to shut down the immune system for some time, and the person becomes very prone to infection. Okay, when we give chemotherapy, bone marrow cells die. Okay, because they are rapidly dividing cells. So cancer patients are extremely susceptible to infections, extremely, because the immune system is dying, because the bone marrow cells are dying. And you know, bone marrow provides all the immune cells, right? So so immune system is dying as a side effect. But in a healthy body, what I understand, what you are asking is that if the body is creating something, then the immune system shuts down for a minute. No, if they're doing everything inside, they don't have to shut down the immune system. If everything is physiological, they don't need to shut down the immune system. See, immune system gets hyperactivated when there is a chance of some danger, some infection. Normally, it is a homeostasis. It is, you know, these um, stuff, these lymphatic system. This is always cleaning up the mess. If it traps any virus, B cells, T cells reside there. Immediately, immune response will be turned on, right? That is why the lymph is always cleaning up all the mess which is there from the cells and everything. So they are not supposed to shut down, because that's what keeps us safe. So immune cells are always surveying us, always fixing stuff. Okay, so the body itself will not shut it down unless it is forced to shut down. Now, here is the thing. Okay, suppose you live here and you start living somewhere else for decades, right? You go to a new place, and that new place is extremely clean. Like, I don't know which place in India is like Darjeeling or some hill station, and you're pretty isolated there, and the air is very fresh and everything is very fresh. So then you come back to Delhi. So now Delhi will attack you, because even you have memory cells, but they may not be that there. So uh, you may get a lot of these things. So immune system became very quiet there, because it had no challenges there, because you were in a very clean environment. But then you come out there, and then the immune system is hyperactive. So there is a joke, there is not a joke, but it is a reality. So when a child is born in US, right? So usually both parents are working. So they keep the child in daycare, right? So the first thing which happens when you put your child, so suppose 3 months you have the child in your home. Sometimes your family member is visiting, so you can have the child in your home for 1 year. Then the family member leaves, and now you have to send the child to daycare. So this is always, every single time, 100% of the time this will happen. So the child goes there, and there are 50 other children there, or 40 other children there. So they bring all different kinds of infection there, right? So your child will get something once in a while, and then very soon your child will be fine after 15 days. And now every single day you are getting cold, because your child has developed a strong immunity. Like 1 year baby or 6 months baby or 3 months baby has stronger immunity than you have. So you are taking care of your baby, the baby has no cold, no rash, and you are coughing every single day. Next day it's a new bimari, and third day it is a new bimari. So the child has developed strong immunity, and then the parents will develop immunity because of the child. So the child becomes a carrier but does not show any symptom. So my point here is that if you live in a very sterilized environment, your immune system will become extremely quiescent. But if you challenge your immune system, then it will be more active, right? So that's why people say that if you're coming from outside, don't drink outside water, because you don't have the immunity for that. uh, Manish was telling me, you have to let the children play outside so that they develop immunity, and that is actually true. But you don't want to challenge the immune system to that extent that it is hyperactive all the time. And nobody does, but I'm just saying that.
As I can understand, I mean, in concern to this, uh, they do immunomodulation or just silencing for a bit is because before tagging them as self cell, they don't want rest of the body.
Oh, absolutely. If you're playing with that, if you're giving a therapy, or if you're doing regeneration, if you're doing organ regeneration, you have to quiescent. So that's what I'm saying. You can shut down the immune system under those conditions where you are giving something foreign to that. Suppose you're even giving CAR T cells, right? You don't want body to react very strongly, but you also don't want CAR T to attack your body. So if you fix both things, if the immune system is slightly silenced, it is good at that time. So you can do that. But the thing is that anytime you are doing that, people who are taking new organs, right, like organ transplant, they are really protected from people, really protected from other people, because they are very prone to infections. People who are undergoing whatever severe medical procedures on a daily basis, their immune system is really compromised. So these people can develop life-threatening condition in no time and die. So you don't want to make the immune system shut down for very long. It has to be very short period.
I get it, but I was talking at cellular level at the very beginning.
When you're doing the therapy.
No. At the very beginning, as in the cell starts. I mean, it's
Oh,
more of
you
a
mean the
developmental
zygote
stage.
is,
Yes. So
you
there's
know, I
just a
don't
quick
know
gap.
how embryogenesis occurs, but I'm thinking
Yeah,
immune system develops pretty quickly.
yeah, because in development biology we do study this, and this is the part where I found a bit of gap.
Yeah, I don't know what time, yeah, I don't know that exactly when the immune system develops or anything. Yeah, I don't know enough about embryogenesis to know what stage what is developing. I know more about the brain when the brain develops because I work
I
on.
know half of an answer of yours because when the inner cell is developing, there is an involution of cells, right? They become the dorsal lip of blastophore, and that invagination, because of animal and vegetative growth, causes the distribution of cells, and then determinants are followed up. That decides what cell become what cell.
Thank you. Thank you. Very good. All right, tea time. No, I checked it. So when actually, Are you able to follow the course? Yeah, a little bit. So I know you have attended quite a few lectures, right?
Yeah,
And
yeah.
some you had to miss because you had to present stuff in
Yeah.
your lab and
Yeah.
all those things. But suppose you have to, suppose you have to deliver a drug to a patient, right? So, like I know you are also very excited about oncolytic virus delivery, right?
Yeah.
So, and you have learned a lot of other things. So, let's say a patient has some pain in, let me think where, some pain in the stomach. How will you go about it as an oncologist? What will be the things you will do?
Okay, I think the first thing is to assess what is the cause of the pain.
Yes.
So, okay, you need to either to check some of the biomarkers.
No, so first we don't even know if it is cancer, right? It could be, so if it is a stomach pain, we'll say, oh, what did you eat? And
Yeah,
we'll do
yeah.
all those kind of
Sure.
digestive things, right?
Yeah.
The pain still persists. Then obviously we'll do sonography to see if it is appendix or something like
Yeah.
that, right? So we'll do that. Everything comes negative. Then what is the first thing you will do to see whether it is cancer?
Okay.
How will you determine if it is cancer? Because if it is deep inside the body, we cannot see the tumor, right?
Sure.
So what is the first thing we will do there?
Okay, if it is a persistent pain, I think first thing you do is to check the biomarkers of cancer.
No, you will check the biomarker. So you will check biomarker when you, first you have to determine, so it could be something else, right? It could be appendix there, right? It could be some digestive issues. So suppose you suspect it is a cancer, right? What will you do there? So let me tell you something. You can do some blood test also, and liquid biopsy also.
Endoscopy.
Huh?
Endoscopy.
You
Also.
can do endoscopy. Very good. You can do endoscopy there. But suppose it is outside of the digestive system, somewhere else. Maybe it is lung cancer or something, right? So many times you do blood chemistry, just blood chemistry, and it will tell you about leukemia and all of those things, right? So a lot of things will come just by testing the blood, right? Sometimes some markers are up and you know that that organ isn't working. So we do all lipid panel and these kind of things. So that can tell us a lot of things about that. But now you don't know what is going on, right? People who have, so then how will you know whether it is cancer? Suppose blood thing is inconclusive. Where will you go next? How would you find it is cancer? You will look for biomarker later on once you determine it is cancer, right?
Okay.
PET imaging, PET scanning. So you will give glucose radioactive, and cancer cells eat glucose a lot more than other cells. So if that lights up, that's probably tells you there is cancer. And then suppose you identify it's lung cancer. What is the next thing you will do? Anyone? This is lung cancer. So let's go step by step. If it is lung cancer, you will ask the person, do you smoke? Are you exposed to secondhand smoke? Where do you work? Are you living in a polluted place? What about the water thing? Exactly. We will check all the things. What kind of water are you drinking? If it is liver cancer, we'll ask, do you drink alcohol? Did you go outside? Liver cancer can caused by lots of viruses. Did you visit some place where there is a possibility? Do you work in the hospital, right? Did you get exposed to something? Did you go somewhere, right? Sometimes people visit mines and things like that, and you know, or you can go to the areas where there is something there.
Long-term exposure to NSIDs,
Yeah, long-term exposures are also bad. So you will actually find out the history about that patient, right? To find out exactly what is going on. So suppose he says, oh, I don't smoke, but I work in an area where people are smoking all the time, like in bars, right? So then secondhand smoke. So then you see that this guy has lung cancer, and then you also know that he's exposed to smoke. So you can make some educated guess about kind of thing, but what will you do next? So suppose there are many kinds of lung cancer. So now you will go to the biomarker to see exactly what kind of lung cancer it is. So how will you find the biomarker? Like, how will you go about the biomarker? Will you do a biopsy of his lungs? Then don't worry about it. There is nothing wrong or right. Just, I'm not a doctor, right? So I'm not an oncologist. So just suggest something. So obviously you can do biopsy, right? But if the thing is spreading, instead of giving, huh? You can. It will be very invasive process, but you can. But you can also, suppose it is HER2, you can give a drug which is fluorescent for HER2, and you see whether it lights up. Sorry, HER2 is breast cancer, not lung cancer, but EGFR is lung cancer. So, and you can do biopsy, and it will tell you what kind of lung cancer it is, what marker it is, or you can give a drug which will bind if it is overexpressed EGFR. So that will give you the thing, and then you can do the targeted therapy, right? And you can, and also, you know, immunotherapy is very good, but it is also known that immunotherapy works best for these kind of cancers versus this kind of cancer. So it's not across the board you will give immunotherapy. You will see what kind of therapies have actually worked for lung cancer, and then accordingly you will give the regimen. You will probably also give anti-angiogenesis therapy, right? So that's how you will decide. And obviously we have to keep an eye on the tumor, correct? Now, yeah, so suppose breast cancer expresses HER2, right? So you can make a small molecule which binds HER2, and you can make that fluorescent. You can attach to a fluorescent molecule, and you can intravenously inject it, and you can image it and see if it lights up. Not fluorescent. Fluorescence will not work in the human body, but some kind of radioactive tag or something. See, radioactive tag is very bad for you, right? But you can give in moderation, because it is, you don't have to do surgery in that case, right? No, we don't want to induce tumorigenesis, but tumorigenesis can be induced by radiation therapy. So this lady I was telling you about, she just went irradiation to kill thyroid cells 100%. And then they have come to me also. My doctor said, oh, I have thyroid thing. She said, oh, you should get irradiated. I said, no. I told to myself that she doesn't know what she's talking about. Because when you do, so I said to her, you know, when you do irradiation, there is a chance you will turn it to cancer. And she sheepishly said, yes, you can turn it to cancer. That is okay. Levothyroxine is pretty non-toxic, and you can take, no, lifestyle doesn't change thyroid that easily. See, the thing is that My cousins in India, they have sent me few things here and there that if you do that, your thyroid will improve. But it didn't improve for me, and it didn't improve for them either. So levothyroxine, it is a pretty mild drug. So if they suggested something, that's not a bad idea. It's okay. It's okay. One thing is there that, you know, if you take a drug, or supplement, even if it is lifelong, it's not a bad thing. Some compliance is very important. Okay, I know of a person here in India, so she has a problem with compliance for the medicine. So she said, not everything gets cured by medicine, and she won't take it. But if you take it properly, it's okay. I mean, some medicines are bad, some medicines are not bad. You can take it lifelong. Metformin for diabetes, it's not a bad medicine. It's anti-cancer. So that's a good medicine. Side effect is anti-cancer. Correct? So levothyroxine is one of the mildest drug. If you know of some exercise of thyroid or something, that's a different thing. I mean, I don't know what she sent me. She said thyroid can improve by that thing, and I'm sure if you check on web, they will tell you do this, that, and thyroid increases. And one thing we did is that we were drinking water from the tap, and the water was contaminated. I mean, obviously it tasted good, but it was something was not good. So we changed the water. The thyroid problem decreased quite a bit, right? So we changed that part, and we never drink tap water. It's been 10 years now, but it is still there, and I take it part of the life. It doesn't matter. I take levothyroxine every single day. I took it this morning also, so it's okay. It's not a big deal. And not for levothyroxine. There are some drugs you go back once, like really, because it has to maintain the blood levels. But levothyroxine, no. I came from US to India, and I missed a lot of doses because first of all I was in the air and my clock was off, and then I got stuck at the airport, and levothyroxine was the last thing in my mind at that time. So I skipped 2, 3, 4 doses there. It's not a big deal. Even if you skip it, even if you're back by a day or two, it doesn't matter. And you have to get your blood test done regularly so that they can adjust the dose. So, but if you know of something, I would be very interested to know some kind of lifestyle change. Yeah, I am not aware of anything like that. I don't think so. I don't think so. That is a different thing. Cortisol you really cannot control, because if you're in a professional life, there is something. But my cousins are not in a professional life, and they have a very nice life, but they still have that. It could be genetic also, but I'm the first person to have it in my family, so I cannot blame my genetics also. Yeah, pregnancy. That kind of thing. Yeah, it could be. It could be that also. It's pretty. For us, it was water which was messing it up, because water had some contaminants. So it was going up and down in both directions for me. So now it is all only in one direction, but it was going in a really, really bad situation for me. So one time it went so low that it was unbelievably low. So I lost consciousness. So I was hospitalized. I was at work and I was meeting some speaker, and suddenly it happened, and I had to lie on floor, and the speaker was taking care of me. Yeah. Yeah, so, but now it is under control. So that time the doctors were testing it every 1 month for almost 3 years, 4 years. But water changing changed a lot for us. So see, pollution can do a lot of things. But thyroid, even if one has to take over like all life, it's a easy trade-off. Yeah. Of course it does. That's how you gain weight or lose weight. Yeah. Yeah, everything is interlinked.
And also that day, a girl was showing that leukemia in West Bengal, the children are more prone to leukemia. So I checked, in West Bengal, what's happening. They have this arsenic contamination in their water, and arsenic is an oncogen.
Of
And
course.
moreover, in West Bengal, the crops are basically more water driven. So they use some pesticides that act as a oncogen for the children, like gamma hexanes and all. That's why they are more prevalent to leukemia.
Oh my God. Oh
That's
my
the
God. And you are right, because they grow rice, and rice is just
Yes.
water, 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 water.
and
Pesti-
yeah.
Yeah, yeah.
And
Fishery,
moreover,
fishery also, yeah.
most of the coastal regions having the humid climate, and that humid climate have the increased amount of sodium concentration in environment. That's
Oh,
also play an important
that
role.
importance, yeah, that is really bad. I was reading somewhere if you grow rice in California or something, just from special region in US, you should not give those kind of rice to cancer patients. So I don't know any details about it, but I'm sure there is something linked there, the way they are growing those rice there. So that's bad for cancer patients. So
When the person who are exposed to the rice fields, they usually caught the hepatotoxic things, like their liver gets easily damaged. Like in my family also I have seen this case.
because of pesticides,
Yes, my
because
pesticides.
of pesticides. So there is this thing which people used to use long time ago in India, rat poison, right? When rats were very prevalent, especially if you are living in villages and something. So that rat poison causes Parkinson's disease. So you are not ingesting it, but you are exposed to it, and it causes Parkinson's disease. So in Parkinson's model, you actually use the rat poison to mimic Parkinson's disease, to study the pathways, what goes wrong. So pesticides are very toxic, and now I don't know, but I'm sure they're using hands to use pesticide. I mean to throw pesticides. So that's so all everything is going inside.
The residues might
Yeah.
stick to their minds.
Actually, our skin absorbs so much of everything, and so when you digest, a lot of things probably is not even absorbed because something gets destroyed. But here everything is 100% going to the blood. So imagine pesticides with hands are directly going to the bloodstream, how bad it is. It is just unbelievable how much absorption occurs here. Like if you're applying a balm on someone else, but you are absorbing more balm than that person, right? So it's pretty dangerous. Pesticides distribution day in and day out is just really bad. And obviously nobody knew about the gloves or anything that time.
Now
So
also people are not using gloves, they are just simply throwing from their bare hands only.
yeah, and that's very bad. It's really bad. Yeah, so I was saying in between also about oncolytic viruses. So oncolytic viruses, first of all, when they're given, they're cleared by the immune system. So they don't even reach the destination. then if they reach the destination, they are not able to penetrate also. That's the inherent property. So they stay at the surface. So if the tumor cells are really deep, if the tumor is really deep, it doesn't go. So oncolytic viruses, for surprising reason, are more effective in skin cancer because they can cross a little bit of layers but not too much. Now, there is one thing, I don't know if you guys know, but whenever you do tumor biopsy and you take out the tumor, usually tumor is necrotic right in the middle, right? I was telling you yesterday also. So the reason it is necrotic in the middle is that oxygen and nutrients cannot get inside that, and oxygen has, partial pressure pretty high. Oxygen cannot get in these tumors are not this big, they are like microscopic tumors, still oxygen cannot get inside there. Okay, still nutrients cannot get inside there. So when viruses are there, I don't know how much they can travel, but they are not very effective. So right now they are trying to do the mesenchymal stem cell therapy to get them there. So if the delivery is good, then probably they will work. And also sometimes they don't replicate very well in tumor cells. That's another problem. So I was telling you, an ideal situation, they don't touch normal cells and they replicate, but sometimes the tumor is so heterogeneous that they don't replicate in those tumor cells also. So it could be a lot of problems there. So on paper it looks great, virus, and that cleans up the virus. So immune system doesn't let the virus get to the tumor. So that is yet another problem. And if you give it intratumoral, right? So it makes skin cells, you can directly inject virus, and that's why they are more effective, because immune system is not involved there directly. And that's why for, deep down things, oncolytic viruses cannot go there.
So we are able to access the liver. So why don't we just operate it, access the liver, inject it, and then
Oh, you can, this kind of therapy is, it is not one-time therapy. And also, oncologists they don't open up the body. I mean, it's extremely rare, extremely rare. They will not open up to inject oncolytic virus. They won't do that. Because every time you open up a patient, there is a chance the patient will never wake up. So actually, whenever they give anesthesia, you have to sign up the paper, even for small things, smallest thing, you have to sign up the paper that you may never wake up from anesthesia, which is such a common thing. So they will not open up the liver like that, and you don't want to inject it unnecessarily. If you dislodge the tumor cells, that can also cause cancer. Yeah, you don't want to do that. Yeah, it's very, very dangerous. They do surgery when there is a very big tumor and it is not metastatic, and you can remove it, and the, what is it? So why did you write your name last? But there's so many. Oh, this is your email addresses. Oh, I see. Because I know these many students were never in the class. So I teach a class, right? And there everything is automated. So to make the students stay there, at the end of the class, you have to take a quiz. Okay, and that is all automated. So they answer it using their cell phone, and it will register online, and within a second it will be graded. So you don't have to grade. It's a multiple choice. No, it's a multiple choice question, right? So the thing is that, so it is this. It's a bigger than this classroom, like 5 times bigger than that, because at a time there are 110 students there, right? And it has 6 sections. So this whole thing we have made geo-localized. So they can only take it inside the room and not outside the room. But here is the thing, my podium is right there. So if they're standing next to the wall, they can. So every single time we count the students, 89. And then when you start the thing, 97 students are taking it because they are next to the wall there. And people are taking pictures and sending to their work. And Google Gemini is telling them what is answer. You take a picture, Google Gemini will tell in a second. And so we are laughing 89, 97 every single day. Then we realize that the 7:30 class on Tuesday, people are very good because people don't come up at 7:30. So if there are 89 students, then 89 responses. So yeah, see, I thought about that also. So maybe that's also happening. And I know that, but then I was thinking that, it all evens out because, there are four actual exams where you don't have phones. So the four actual 400 word points of exams will filter out the things. So even if you gained one extra point, it's not a big deal. Your honesty will pay at the end of the day. All right, next question, guys.
Ma'am, my question was why do cancer cells why do we observe tumor heterogeneity? What is the utility for the tumor to be heterogeneous other than just evading the treatments or any immune action? Is there any other utility for that?
So obviously it is there. By being heterogeneous, they can survive under many different conditions, right? And it is always survival of the fittest. Second reason they are heterogeneous because it is like they need help from a lot of other cells. So suppose there's tumor cells, CAPS cells. CAPS are cancer-associated fibroblast and all of these immune cells. So they're all participating in making that thing. So that is one reason. Then there are fibrous cells which are protecting the tumors. So it's always synergy between them. They are giving something, they are getting something, right? So they make all of this their own eco environment to survive. Otherwise, cancer will not survive because immune system will eat it up, right? So they make their own colony of people to do that. And on top of that, yeah, so most of the diseases are multifactorial. It's not one thing which is going wrong. So later on, one person may become the king of the castle, but there are many things which contribute. So that also contributes to heterogeneity, like Alzheimer's disease, for example. If I work on CDK5, I will say CDK5 is the culprit. Somebody works on something, they will say that is the culprit. They will say that is the culprit. But the truth is, it is multifactorial disease. So if it is multifactorial, it will be heterogeneous.
Ma'am, but they ultimately end up being an undifferentiated mass. Is that just because they have proliferated in such a manner that they do not need the help of those other cells anymore? Or is it, why do they end up undifferentiated at the end of it?
No. So their main job is to just keep on growing. So they are undifferentiated because if they differentiate, they cannot grow. Differentiated cells don't grow that much. Okay, undifferentiated can go keep on proliferating. Plus they are not organized. So suppose you build a house, right? And you will put the brick on top of brick and brick properly. So that's how the body is built. Everything is properly organized. Why is it a tumor? It's just a bunch of stuff. It's like you're putting brick, throwing brick over brick over brick over brick. So that also creates a lot of vulnerability for cancer cells. So in the last lecture, I will show you that cancer cells, the blood vessels, they have to make the blood vessels. Everything is really fast. So blood vessels are very leaky. So if you give a drug, they accumulate at tumor sites because the blood vessel is leaky there. So it will just go, and this is called EPR effect. Okay, so they will go in there. So tumor cells are a mass of things, but they do need help from a lot of other things. They steal, they change, and they will, they secrete exosomes. Exosomes will have microRNA, and they will make all the innocent bystander cells oncogenic. So they are constantly spreading and spreading and spreading. And the thing is that they kill the person and then they also die, right? So they are very smart, but they are not smart enough to keep the person alive, right?
Yes, ma'am. Could we say that the degree of heterogeneity that we would observe in a tumor would vary from cancer to cancer?
Yeah,
Ma'am,
cancer to
so
cancer.
if I think what I thought was that if it's skin cancer or if it's liver cancer, liver has many specialized cells and its general population is very diverse. Meanwhile, in skin we do not have as many types of cells. So would the heterogeneity be impacted by the local, like the cell environment as
I
well?
would imagine so. I would imagine that. I don't know the details about every
Yes, ma'am.
cancer, but I would imagine that, and it also depends on, like, skin cells require a different conditions, right? And then something which is deep inside the body, they make like a lot of immunosuppressive environment. Pancreatic cancer, for example, this tumor size and the whole outside is this big. So that is very unique to pancreatic cancer. So obviously the microenvironment decides how heterogeneous it is, what kind of things. So it depends on the tissue type, what cells are there, what is permissible there for cancer cells also.
And ma'am, can metastasis be called a purely chance event, or is there a chain of things that leads to
Oh,
metastasis?
it is not a chance thing. It is a whole highly orchestrated process. Okay, so epithelial to mesenchymal transition is one of them.
Yes,
So
ma'am.
it is a real big process that happens to make the cells very motile. There is a whole upregulations of factors which make the cells very motile, and that's when they metastasize. And the cancer cells can, like, really, I did not tell you all the details because it's so much information, right? So I cut down on a lot of information, but the cells will look like they are dead. So they will become senescent like that for a long time. And then the advantage is that you can keep on giving therapy, nothing will happen. When all the therapeutic pressure is gone, they will wake up and they will spread cancer. Cancer stem cells do the same thing. They exit cell cycle because we are attacking cell cycle. They will go to G0 state, come out of the cell cycle, stay there for a long period of time. Then they will just wake up and then do it. So they are live things. They are really live things. You know, see, I will tell you, they obviously don't have brain. So you must be thinking, like I am thinking, how do they know? But if you go, if you grow E. coli, right, bacteria, so you're talking about a single cell there, right? And I'm not even talking about eukaryotic system. I'm talking about the most basic prokaryotic E. coli. So E. coli, what it does, it divides every 10 minutes, dividing, dividing, dividing. If you take out E. coli, it grows at 37 degrees. You take it out and you put it at 100 degrees, they will die because it's like a severe accident and it will die. But if you put it at 42 degrees, right, so it's still very high for them, but they will survive. So how will they survive? These are E. coli, right? No brains, but this is live cells. So how they survive is that they will stop dividing. So they know that the conditions are not optimal, so they will stop dividing and they will use all their energy to make heat shock proteins, because when you heat it up, DNA can get denatured, proteins will get denatured. So they will first make heat shock proteins to save themselves. And once they save it, and once the condition becomes optimal, then they start dividing. Okay, now suppose E. coli, for examples, just like we love ice cream, they love glucose. Okay, so they will just keep growing in glucose. Then you mix a little bit of lactose there, and they will ignore lactose and survive on glucose. Okay, now you remove complete glucose and just give them lactose. Now it is a question of life and death. So now they will turn on the lac operon and make all the genes which are required for lactose consumption. So think about a prokaryotic cell changing so much depending on environmental conditions. So now it should not come as a surprise that cancer cells are very smart. And what is coming out is that there are a lot of pathways which are turned on while during embryogenesis, and these pathways are turned off upon birth. And cancer cells hijack these pathways and turn on these embryonic pathways, which really supports cancer growth. So basically, all the information, like you said, is in the cell and they just turn on. It's all encoded in the cell and they know how to access it. Yeah.
Ma'am, driving it from the fact that our antibodies use multiple times, I mean, various type of permutation and combination just to make that exact fit according to the epitope. I mean, basically the
Yeah,
antigen.
yeah.
So, on the basis of that, even if our immune system gets hijacked by the cancer cells, no, our antibodies get stopped. So even after the modulation of epitope, again, then again our immune system should produce the antibody. Why does it get shut down? It should, I mean, accordingly. So, I mean, if it's changing, then it should change accordingly.
So there are a lot of things which are going on, right? So first of all, it are the T cells. It's not the B cells which are going next to the tumor. It's the T cells, and T cells are changed into Tregs. Okay, or T cells are rendered ineffective, or tumor cells will make the MSC molecule disappear. So T cells are not able to recognize them. Okay, then they will make all the tumor cells like very acidic. So anything which comes there, they are just too tired to attack. So the tumor suppressive environment does not let immune cells come to close to the tumor. And if it comes close to the tumor, they change it to tumor pro-tumorogenic. So that's the bad thing. They make it pro-tumorogenic. They support tumor formation. So basically, they change their affiliation. Instead of anti-tumorigenic, they become pro-tumorigenic. So that's what is happening. And that is why immunotherapy is so good. Now, B cells, they reside in the lymph nodes, right? So if the tumor goes to the lymph nodes and if B cells are there, they can form antibodies, but then the tumor will change the epitope and the antibody will not work. So, see, the thing is that if our immune system can fight it off, the tumor will not grow. If the immune system has upper hand, the tumor will not be aggressive. Okay, for example, in colon cancer, it takes almost 15 years for the cancer to develop. So basically, the immune system is able to fight it off. That's why it takes so long. But in some cancers, they are so aggressive, they can just. Now, for pancreatic cancer, the whole thing is hiding in a whole layer of fibrous thing. So I am very big for this room. I mean, this is like a whole building of stuff is protecting one human being. That kind of fibrous stuff is there. So immune system is not going to even reach there. And the tumor is so shielded, nobody will see the antigen or anything there. I was just looking how nicely she writes.
And again, it's a curious type of question. We were discussing about bisystonic antibodies.
CAR T cell therapy, yeah.
There was a section where we had three hands, right?
Yeah.
So I'm just curious to know, can we use drug in that one arm hold side of it, because drug needs to be delivered in third form. So we do all the activation, all the chemical chemistry in these two hands only, and then immediately deliver drug to the cancer cell.
So CAR T cells are holding on to the drug
In a pro form.
in the pro form.
maybe some
So
inducer.
CAR-T is a different thing altogether. CAR-T is not a delivery vehicle. It's a much easier way. CAR-T is too sophisticated to deliver a drug, and that is not its primary thing either. CAR-Ts are engineered to identify the antigen on the tumor cells and kill the tumor cells by spraying perforin, granzyme, cytokines, things like that, and recruiting other immune cells. So if you have to, so let me ask you this question. You guys, it's open to all. Okay, if you have to deliver the drug specifically to tumor cells, what will you do? You don't have to spend $500,000 to do that. So tell me, there is a whole lecture, I think 29th lecture is on the delivery, 30th lecture is finally deep medicine, which was the title of the course. Okay, but and 28th lecture is polypharmacology. But tell me now, how will you deliver a drug specifically to tumor cells, whether it is in the product form or it is in the drug form?
Using nanobodies.
PROTAC is,
It's a linker.
yeah, PROTAC is not a delivery method. Yeah,
Bringing things reliably to the
no. PROTAC is just binding to a protein of interest and bringing it to E3 ligase, but it's not a delivery vehicle. So, so I'm going to do this thing. I'm going to ask all of you. So raise your hand. Everybody has to come up with one way of delivering a drug. So if you raise your hand, you can say something which other person was going to say before that person. So, okay, you start. Yeah, that is very good. That is one way, like Herceptin antibody. So that's ADC, antibody conjugated drug. Yes, go ahead. You had your hands raised, right? Yeah, go ahead. Absolutely. And then how will the nanoparticle get to the tumor site? Because it will go everywhere in the body, but how will you deliver to the tumor site? Go ahead.
We can make use of the tumor specific microenvironment, like creating a bond which will like if acidic environment is in surrounding, then we can use such a particle which will detach in acidic environment like generation.
And then deliver the drug. Very good. Go ahead.
Using societies.
Yeah, suicide genes, but how will you deliver it to the tumor site?
Ma'am, like the specificity will be there. Like if we, for example, you have given an example of HSV-PK1 enzyme
Yeah.
that is incorporated with ganciclovir or something like that, right? So that will be target specific only, like it will bind to.
No, 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 no. So if you give the drug, it is systemically going through the whole body system, and then it needs cytosine deaminase enzyme. How will you deliver cytosine deaminase enzyme to the tumor? Otherwise, the drug is ineffective, right? So when you give the drug, it will be converted to 5-fluorouracil. If cytosine deaminase enzyme is there, how will you deliver cytosine deaminase to the tumor site? How will, no, your turn is over. So everybody has to speak up. So anyone, can you tell me how can you get cytosine deaminase to the tumor site? I told you today also. Venkatesh, Vanshika, you have already said, so I won't ask you. I've asked Evans a lot of questions already. Unless you want to answer, what about you? You have already answered. Yeah. What is your name? Divya. Divya, right? Yeah. So Divya, can you tell me how can you deliver it? How can you deliver cytosine deaminase to the tumor site? What genes? See, think about it like you think of a story, right? So don't think of it as like the way is that you think tumor is there at some site of the body, right? And you have to deliver it to the tumor site? It's open to all. Anyone wants to answer this question? Yes.
One more microenvironment-based approach that in morning you said this tumor cell secrete VGAF and CHCI-IT. So we can use that. Also, like in case of mesenchymal stem cells.
That is the one. Great. Got it. Mesenchymal stem cells naturally go towards the tumor site. If you put mesenchymal, they will find their way to the tumor site. So in the site of mesenchymal cells, you put the DNA of cytosine deaminase. It will be delivered to this. This is a fantastic way, right? This is a fantastic way to deliver it there. Now, so tell me some other delivery method. Anyone? Nanoparticles, she has said already. Any other way? You haven't said it as yet. Okay, that is that is a kind of cheating, but we can do that. You are right. Nobody said it, so you get the points for that. If it is at least skin or something, you can do it. Okay, good. That is doable, right? Car is too expensive to deliver the cargo. And cars are not meant for that thing. You will not use car for that. Oncolytic duplicate of HER2 cancer cells. It's a small. Again. The names because I breathe. Can you think of some? The names because I breathe. Can you think of some? If it is right, so you don't want clue here. If it is right, so you don't want clue here. Malade part 1. And so sometimes I hear nanoparticles and I said so sometimes I hear nanoparticles and I said
Lies as
in way is that added to it. The whole cell way is that added to it. The whole cell is full of
okay, so that
do that. Cell is full of
okay, so that
do that. You have to answer it too. Cyto, you have to answer it too. Cyto to travel the. It's like great travel the. It's like great the hook right now. Not meant to be for the cargo. It's a cell which is awesome. Coated with four the nanoparticles. So they with four the nanoparticles. So they will be taken to something like that. Ignition signal on NANOS remains active. That ignition signal on NANOS remains active. Everybody in adding the drug to yes, absolutely. In adding the drug to yes, absolutely do that. Absolutely do that. First-stranded DNA. These what will bind. So this will or at least target of interest and what were binder, which will bind the target. What were binder, which will bind the target cells. So you can take oncology, but in realizing so you can take oncology, but in realizing so I met some rainbow diet that like all the color rainbow diet that like all the color set IIT Kanpur